Hello everyone! Today we're doing a really long biology review. This video is a compilation of many of my other biology review videos, and I put them all together in one session, so if you really need to cram or just get a lot of biology content out of the way in one sitting, this video is for you. Now this video is designed to review biology concepts for a high school biology honors level course. Depending on where you live and who your teacher is, some of the content in this video may be a little high level for you, or you may have never seen some of these concepts. There also may be content in your course that you studied that is not covered in this video, but I did my best to include some of the most important information and topics that you might need to review an entire biology course in a little over 2 hours. Now, as you're watching this video, be sure to take breaks, get up, move around. Please don't do this all in one sitting. If this video is helpful to you and your studying, please make sure you give it a like, and without further ado, let's get started. So water has some really unique properties that make it a really essential, important resource to living things. It's often called the universal solvent because it can dissolve a really wide range of substances, and this makes it vital for the transport of nutrients, gases, waste molecules, and it makes it a great place for chemical reactions to occur within cells. In different organisms, 70 to 95% of their structures, of their body structures, is actually made up of water. And in humans, water's unique properties contributes to homeostasis and our survival as well. Water moves in and out of membranes through osmosis, moving from a high concentration of water to a lower concentration of water. In humans, water is a thermal regulator. It helps us regulate our temperature. It helps, obviously, transport materials. Our blood is composed of water, but plants too have plant sap, which is composed of water. It cycles through us. We eliminate extra water through our kidneys. And in our bodies, the liquid water is the most important, but it can also exist as water vapor and, of course, as solid ice. So let's talk about those properties. Water is a polar molecule, which means it has an uneven distribution of charge. So one side is more negative and one side is more positive. The oxygen atom is more electronegative than the hydrogen atom, so it pulls electrons more towards itself, which gives that side a partial negative charge, and the hydrogens have then a partial positive charge. And because of this positive and negative side, this leads to hydrogen bonds, or this attraction that happens between water molecules. So remember, it is a solvent. It can dissolve a lot of different things, but because of its polarity, it can stick to itself really well. That's cohesion. It can stick to other substances. That's adhesion. It forms really strong attractions to itself. When there's a layer of water molecules on the surface, it's more attracted to itself than the air above, so that contributes to surface tension. It also has a high specific heat capacity, which means it resists changes to temperature, which means it can absorb and store a lot of heat before its temperature rises, and that is crucial for temperature regulation in organisms and helps us maintain a stable internal environment. It also has a high heat of vaporization, which means it takes a lot of energy to convert from liquid to gas. And so in cooling processes, and there's liquid sweat on our skin, the evaporation decreases the temperature of our body by absorbing some heat during that liquid to gas phase change. So let's add a little bit more vocabulary for review. Again, polar, meaning water has a slightly positive and a slightly negative end. It'll work well with other polar molecules, and non-polar substances will tend to avoid water. So molecules that are water loving or that interact and are soluble in water are often called hydrophilic, and molecules that are water fearing, hydrophobic, are insoluble in water. So we think about the structure of our phospholipid bilayer in our cell membranes. Those hydrophilic phosphate heads are going to be more attracted to water, and the hydrophobic tails are going to avoid water, and so they'll situate themselves in the center of that membrane. Now, like I said, cohesion is the attraction of water molecules to each other. Adhesion, thinking you're adding another substance, it's the attraction of water molecules to other types of molecules. So we can see how water beads with itself on surfaces. It touching those surfaces is adhesion, but that beading and droplet formation, that's cohesion, because the water is connecting to itself. And even if you watch water fall down a window, you can see the droplets of water that gather together to form larger droplets. That's cohesion. Capillary action we often see in plants when we have cohesion and adhesion working together up the stems and xylem against gravity. And of course, in surface tension, again, water is more attracted to itself than to the air or other things around it. That's why things float on top of lakes, or certain bugs can crawl on top of water. This is due to surface tension. Now let's touch briefly on molarity. This is more of a chemistry concept, but we'll touch on it in laboratory settings in biology. Molarity is a measure of the concentration of solute, the thing that's being dissolved, in a solution. It's expressed as number of moles of solute per liter of solution. solution, so moles per liter. So usually we'll see it in the context of a lab when we have a particular solution. We'll see the molarity of a specific chemical or molecule in a biological sample. It's good to recognize that something with higher molarity has a higher concentration of particles within it, whatever that substance may be. So something that's 10 molar is probably going to be way stronger than something that's 1 molar or 0.1 molar. So a 10 molar solution would be strongest, 1 molar would be relatively intermediate in this situation, and 0.1 molar would be the weakest in this particular spectrum. I'm going to go through the monomers of each of our main categories of organic compounds or macromolecules, talk a little bit about their function, and then some indicators which you might use in the lab or you might have used in the lab to identify each one. So nucleic acids are made of nucleotides, that's our monomer or single unit to build our larger things like DNA and RNA. They consist of a phosphate, a sugar, and a base, and then many of them link together to carry genetic information in organisms. Proteins, which DNA code for, DNA actually carries the instructions to build proteins, are made of amino acids. Proteins do all the jobs within cells and living things. They transport materials, they provide structure, they send signals, they receive signals, and they are also enzymes which help catalyze or get chemical reactions going. So they do all the jobs of the cell, and all of our genetic traits are based on proteins because that's what our DNA codes for, is proteins. Now these amino acids have a very specific structure, which we'll talk about in a moment, but they have one really important piece called an R group that is on one side of each amino acid, and that piece varies from amino acid to amino acid. So different characteristics of that R group are going to give the amino acid different properties, which then changes how the amino acids will fold and link up with each other. So we get these long polypeptides or these long chain of amino acids, and every amino acid influences how the protein is going to fold and its final structure and then its function. Carbohydrates, like I said before, are made of monosaccharides like glucose, which is a simple sugar, and then larger polysaccharides are built from those monomers all put together. So many glucoses put together will give us cellulose, which provides structure like in the cell walls of living things, but on its own, carbohydrates are responsible for energy. So we use glucose in cellular respiration and in fermentation to provide energy to living things. So that's really important as far as function goes. Lipids are going to be made of fatty acids, triglycerides, but one example that we see in every living thing are our phospholipids, which are what the membrane is composed of, the cell membrane. So we have a phospholipid bilayer. That phosphate head is actually hydrophilic. It is attracted to water. The lipid tail is hydrophobic. So the way the cell membrane arranges itself is due directly to the properties of this particular molecule. Now, lipids can also be used for long-term energy in living organisms. They're not only in membranes, and they can also provide insulation like blubber in mammals. Now, we use different tests in the lab to identify these organic compounds or biological macromolecules. For example, DNA, you can use gel electrophoresis to see different banding patterns of DNA. We'll talk more about that when we get to biotechnology. Biuret reagent is a common chemical used to identify proteins. It turns this pretty purple here when it encounters peptide bonds, which are the connections between amino acids. Benedict's reagent is another chemical we can use to identify carbohydrates, simple sugars. So if there's something like glucose in a substance, we can add some Benedict's, heat it up, and it'll turn this pretty orange. It originally starts out as like a blue. And then test strips, you can also use glucose test strip to identify our simple sugar glucose. And then iodine is used for things like starch, so longer carbohydrates. Remember, starch is a carbohydrate too. And then, of course, we have our very fancy brown paper bag test to identify lipids. All right, one thing these things all have in common, despite their different functions and structures, is they all contain carbon. That's why they're called organic compounds sometimes. And they also contain hydrogen and oxygen. Now, they have other elements present in them as well, but they all contain carbon, hydrogen, and oxygen. Let's take a closer look at proteins really quick before we move on. Remember, proteins are long chains of amino acids, and each amino acid is one monomer, one subunit, and it has that carbon in the center with the hydrogen attached to the top. And remember, carbon can make 4 bonds. One of those will be to the hydrogen, one of those will be to what we call an amino group, so that N and 2 H's, and one of them will be to a carboxyl group. Now, the protein will grow by attaching more amino acids on the side of that carboxyl end. But then the side chain, that R, isn't just an R, that's not an element, that is just a placeholder for whatever other molecules are going to attach to the amino acid and give it its properties, like maybe being hydrophilic or hydrophobic or ionic and more acidic or basic. And so every amino acid has different characteristics that are based on the side chain or this R group. DNA, of course, is super important too. If we go back to nucleic acids as one of our categories, and just like water, in the center, it's connected by hydrogen bonds, which can easily break and then come back together when we need to replicate the DNA or undergo protein synthesis. And you can see all of these nucleotides built together to form the structure of our DNA double helix. So different cells will have different organelles depending on the functions that they serve. Prokaryotic organisms are going to have different organelles than eukaryotic organisms. And remember, prokaryotes are organisms that lack a nucleus and membrane-bound organelle, so they're going to be relatively simple compared to our eukaryotic organisms. They're going to be pretty small, and in general, they will have fewer components. Our eukaryotic organisms, including plants, animals, protists, fungi, these will all have cells with membrane-bound or organelles that are surrounded by a membrane. And we can compare different types of cells like plant and animal cells, both eukaryotes, and see, for example, that a plant cell has a chloroplast and an animal cell does not. So let's look at these two pictures. We have the nucleus drawn simply here, plasma membrane, which is the surrounding double layer around the cell, which if we zoomed in closely, we see is a phospholipid bilayer. The cell wall with plant cells and certain prokaryotic cells, and mitochondria, this oval with the squiggly line, our vacuoles here and here, chloroplasts, again only in plant cells, and ribosomes, represented frequently by little dots. Sometimes you'll see a mitochondrion drawn very technically, and they look a little bit more like this than this simplified drawing here, but sometimes you'll just see an oval with a squiggly line. So you need to be able to recognize both types of pictures. And you'll also potentially see a chloroplast drawn more like this than the one I have in my picture. These are stacks of thylakoids, which are an important part of the chloroplast. Don't really need to focus on that for the biology EOC, just be able to recognize chloroplast as an important organelle with these kind of stack-like structures within a plant cell only. So getting back into the differences between animal and plant cells, which you might remember again from other science classes, plant cells have cell walls, and they look kind of like these geometric shapes. Animal cells do not. Plant cells also have chloroplasts, where animal cells do not, and plant cells tend to have one large vacuole for water storage, where animals can have smaller vacuoles or multiple vacuoles. So make sure you recognize those main differences between plant and animal cells. Those aren't the only differences, but those are key important ones. So recognizing our organelles by picture, again, here's our simplified mitochondria. This is for energy, so you need to be able to understand that the mitochondria is where the cellular energy is created or made in a process called cellular respiration. You need to know more than just the mitochondria is the powerhouse of the cell. Here are simple circles with kind of a blank space in them. That would be a vacuole, which again is for storage. Our ribosomes are represented by little dots, and this is where proteins are made. Our nucleus is going to be in the center of the cell. Sometimes you'll see chromosomes if the DNA is condensed represented in them, but it's going to store our DNA or our genetic information in eukaryotic organisms, and we'll get to that in a second. Our cell membrane is going to be represented, if we zoom in closely, by this phospholipid bilayer, and its purpose is to provide a semi-permeable barrier, letting some things in and some things out of the cell in order for the cell to maintain homeostasis and do everything that it needs to do. Our cell wall is going to provide structure and support, another layer of protection of the cell, again, not in animal cells, and it's going to be kind of geometric in shape. And our chloroplast is the site of photosynthesis, only in plant cells, so we're not going to see these in animal cells. Now, if we look back at our plant and animal cells, these are both eukaryotic organisms, meaning they have a true nucleus, or they have a nucleus and other membrane-bound organelles. So organelles like the mitochondria, organelles like vacuoles, and these are generally larger and more complex. Prokaryotic organisms are more primitive. We think they evolved first. They typically only have a few key features. So they're very simple, they're very, very small, they're more abundant on Earth than eukaryotic cells, but they're very different. So in prokaryotic cells, we do not have any of those membrane-bound organelles. We don't have a mitochondria, we don't have vacuoles, we don't have a nucleus. Instead, the DNA is just free-floating within the cell in a feature we call the nucleoid. We do have ribosomes because all cells do make proteins and have to do protein synthesis. We do have a cell membrane to contain the cytoplasm and the essential functions of that and the essential features of that cell. And that's about it. Sometimes there'll be external features like our flagella, which is for movement, or cilia, also for movement. Those are small hair-like structures. Those can exist in prokaryotic organisms, but again, not all prokaryotic cells have those. So again, prokaryotic organisms, much more simple, no nucleus, no membrane-bound organelles, very, very simple features. Plant and animal cells are both eukaryotic. A few other organelles I wanted to mention include the endoplasmic reticulum. We both have the smooth ER and the rough ER. The rough ER contains ribosomes. And this organelle is a network of tubules and sacs that helps transport proteins and other materials within the cell. This right here could be a lysosome, maybe a peroxisome, but it's a lysosome that is a membrane-bound sac that has enzymes that helps break down waste products and other damaged organelles, kind of like a garbage disposal of the cell. And then, of course, the Golgi apparatus or Golgi body that is responsible for packaging and shipping materials within the cell or out of the cell. Now, that is just a quick glimpse at some important organelles, the basics. Obviously, there are more organelles you probably encountered or will encounter in your biology class. Remember, all cells, no matter which type of cells, are going to be surrounded by a cell membrane. They're also going to have genetic information, DNA. They're also going to have cytoplasm, that gel-like substance where all the components of the cell sit. But the membrane is really crucial for containing all the important materials of the cell. So as we mentioned in other videos, the cell membrane is a thin layer that surrounds all cells. It's made up of a phospholipid bilayer. So those phospholipids are lipids, and they have phosphate heads, which are hydrophilic, that will interact with water well, and then tails, two lipid tails, which are hydrophobic, that tend to avoid water. They are water avoiding or water fearing, according to the name. The way the bilayer, the phospholipid bilayer, arranges itself, and right here we're just looking at a cross-section of a membrane. Remember, this is all the way around the cell. So if we were like zooming in on one single section of our cell membrane, this is what we're looking at. But the outside of the cell will have the heads facing outward, and then the inner components of the cell will also have those phosphate heads facing inward too. So the tails arrange themselves in the center of that double layer. These pink dots we will come back to. Right now they're just representing different particles that could be outside or inside the cell. Now remember, all cells, whether eukaryotic or prokaryotic, will have a cell membrane, that phospholipid bilayer. Some eukaryotic organisms and some prokaryotic organisms may also have a cell wall that is an extra layer of protection and support that surrounds the cell membrane. But all cells, no matter what they are, even if they do have a cell wall, do have a membrane as well. Now let's dive a little bit deeper into some of the other things that are happening within the cell membrane and what makes it so special. Now it's not just phospholipids, even though that's sometimes what are depicted, and it's not just transport proteins, which you might see in diagrams, and we'll get to those in a moment as well. There's also other integral proteins, peripheral proteins, carbohydrates, cholesterols, and each of these serve a different purpose within the cell membrane. We call it a fluid mosaic because there are lots of different pieces and components. That's the mosaic, and then fluid because it is fluid. There is motion to it, it's flexible, and one of the most important molecules in the brain that is going to help stabilize it but also regulate the fluidity is the cholesterol molecule, which is a lipid as well. And we can see where it's found within that bilayer, again towards the center, since lipids in general are hydrophobic. Another important molecule in the membrane you might hear about are carbohydrates, which primarily we will see on the extracellular side, so on the outside of the cell membrane, and they're used for various purposes, from protecting the cell to signaling to other molecules to serving as identification markers on the cell. A lot of times we'll find them attached to glycoproteins, and there'll be other proteins as well that can be found on the outward-facing side of the cell membrane that are going to help for signaling. They're going to help with identification of the cell or help the cell adhere or stick to other cells. There's a lot of different functions that these membrane proteins could have. Now, often when we talk about proteins within the cell membrane, we are talking about transport proteins, which are going to allow certain molecules in and out of the cell. Now, the cell membrane serves as a protective barrier. It helps it communicate with the environment, but it also helps regulate what goes in and what comes out of the cell. And that's where sometimes these transport proteins get involved. So let's take a look at how molecules can get in and out of a cell, starting with passive transport, or transport that doesn't require any additional energy. So first of all, there's simple diffusion, and these are really small molecules that are able to go from high concentrations to low concentrations without any extra energy added, and they don't need a special pathway or a door, quote unquote, to get through the membrane. We'll see diffusion happen with nutrients and gas exchange, so the diffusion of oxygen into the blood. But if we're looking at a diagram, if we see molecules moving from a higher concentration to a lower concentration and there's no protein, no ATP, then we can identify it as simple diffusion. Now there's also facilitated diffusion, which often occurs with larger molecules or molecules that aren't able to interact with the cell membrane because of their charge. And they will also go from a high concentration to a low concentration, high outside the cell, lower inside the cell, or vice versa. And no energy will be needed for this process, but they'll use these either carrier or channel proteins to get inside the cell. Again, both of these are passive transport. Finally, we get to active transport, which does involve ATP or energy. And this is when we are moving from a low concentration to a high concentration. So we're going against the concentration gradient, and molecules are being sent in a way that requires extra energy. And we're going to use an extra transport protein to have this happen. A good way to remember the difference between all of these is I like to think of the slide metaphor. So with simple diffusion, you're like a kid just going down the slide. You're going from a high point to a low point, so think high concentration to a low concentration. And when you go down the slide, think you're not using any energy, you're just falling down the slide. Physics people, don't get mad at me. We're just going to ignore kinetic and potential energy for right now. Then in facilitated diffusion, you're still going from high to low, going from the top of the slide to the bottom of the slide, but you're getting a little help. So think of it as a child who's getting a push down the slide or having a parent that's helping them go down the slide with them. Then active transport across the cell membrane is when a kid is climbing up the slide, like we were all told not to do when we were kids. We are going from low to high, so think low concentration to high concentration, and for that kid, it does require them to put out some energy. It's tough climbing up the slide. Anyway, it's a simple mnemonic that I like to use to remember the difference between all these different types of transport. All right, one last type of transport that I wanted to mention for today is osmosis. Now, osmosis, simply put, is the diffusion of water. So it's when water moves across the membrane from a high concentration to a low concentration, and often occurs when there are differences in concentrations of solute, so substances dissolved in solution that can't cross the membrane, but water can. And in general, water will move towards where there is more solute, even though the water itself is going from a high water concentration to a lower water concentration. So let's look at a few examples. If we have a cell and the concentration of particles in and out of the cell is relatively the same, that's called an isotonic solution, equal on both sides. And so we're going to have a net water move. Water is still going to move in and out, but it will do so at approximately an equal rate, and so we won't have any net gain of water or net loss of water out of the cell. Now, if we have a situation where there's actually a higher concentration of solute, of particles inside the cell, and a lower concentration of solute outside the cell, what's going to happen is we have a hypotonic environment. Think hypo meaning low, so low solute. And if we think of the term solute sucks, we can remember that water is going to move towards where there's a higher solute concentration, but think also about where there's a higher water concentration. Remember, water itself will move from where there's a high water concentration to a lower water concentration. So there's a higher water concentration, apparently, on the outside, a lower water concentration comparatively on the inside, so water will move into the cell. Often this may cause the cell to expand or even burst, but that depends on the situation. Now in this environment, we have a higher concentration of particles outside the cell, a lower concentration of particles inside the cell. This is called a hypertonic environment. Think hyper, like excited, like there's a lot, so there's more particles outside the cell. And again, water will move from a higher concentration of water to a lower concentration of water. So in this case, it will move out of the cell. The cell may even shrink. So let's do a little bit of practice together. A cell is in a beaker with a solution with a higher salt concentration outside than inside the cell. What will happen to the cell? Think about it. Water is going to move out. Remember, solute sucks. Water will move towards the lower concentration of water and out of the cell, and the cell may even shrink. A cell is in a beaker with a solution with a lower concentration outside than inside. What will happen to the cell? Well, pretty much the opposite. Water is going to move in, the cell may swell or even burst. Okay, now let's put some numbers to it. A cell is in a beaker with a solution with an 85% salt concentration on the outside and a 15% salt concentration on the inside. Salt cannot cross the membrane. What will happen to the cell? Now think about it in terms of numbers. So we have 85% salt on the outside, 15% salt on the inside. If you're doing this on your own with no diagram, you could even draw it out if you had a problem like this. And then on the outside, there's 15% water, 85% water on the inside. So where is the water concentration highest? It's on the inside of the cell. Water will move from high to low, so water will move outside of the cell. What could happen to the cell? It could shrink. All right, finally, our last one. A cell is in a beaker with a solution with a 25% salt concentration outside and an 80% salt concentration inside the cell. What will happen to the cell? Think about it. Water will move in and the cell could swell or even burst. So enzymes are extremely important molecules in biology. They are going to be catalysts that help certain reactions occur. They can help break down certain molecules or put other ones together. They're important in photosynthesis and cellular respiration and digestion. In almost everything that's going on in the cell, enzymes are there and they are key. We'll talk about enzymes in medicine and health. Think about people who are lactose intolerant. It's because they don't have enough lactase enzyme to break down lactose, that milk sugar. In the environment and ecology, we'll continue to talk about enzymes. And remember that enzymes help reactions happen. Now, one key way to recognize an enzyme, even if you've never seen it before, is the letters ASE. Now, not every enzyme ends in the letters ASE, but many do. Even things like Rubisco, their full name is a word that ends in ASE as well. Let's take a closer look at a basic enzymatic reaction. So we have an enzyme, which remember are proteins. We'll get back to that in a minute. So we have this specific configuration of amino acids with one part called an active site where it's going to bind to a substrate or the molecule that is going to undergo the reaction. It's the reactants. This substrate will then attach to the enzyme and make what's called the enzyme substrate complex. The reaction will occur and then the products will be released. The enzyme is unchanged and can complete the reaction again and again. We call enzymes reusable because of this. And if you have enough enzyme to complete the reaction, then it'll keep going and going until there is no more substrate to act on. Now, let's get back to this idea that enzymes are proteins. So remember our four main categories of biological macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Enzymes are in the proteins category. And it's important to remember that because it's important to remember what enzymes are made of, and that is amino acids. So let's take a look at our protein structure once again. So because enzymes are made of a long chain of amino acids, they're joined together by peptide bonds, and they'll have a secondary structure too with connections between different amino acids through hydrogen bonds, and then of course a tertiary structure that gives it its three-dimensional shape. And the shape is very important because it determines the active site where the reaction is going to happen with the substrate. Now, just like any protein, if the conditions are unfavorable, let's say things get too hot, those bonds can start to come apart and this enzyme can lose its shape. So it unfolds or denatures, and then we may not have an active site that's the same shape as it should be to interact with the substrate. So denaturation is really going to affect the reaction rates because the structure of the protein is no longer there to be able to fit with the substrate and for it to attach and for the reaction to occur. Now, there are other ways that enzymes' activity can be affected as well. It's not just temperature. This can be pH. This can be the amount of enzyme there, the amount of substrate there, like we already mentioned. It could also be the presence of inhibitors. So inhibitors are other molecules that can either attach at that active site and block the substrate from binding, so that's called competitive inhibition, or they can attach at other points on the enzyme at an allosteric site, is what it's called, and that might cause some configurational change in the enzyme as well and cause the active site to no longer be compatible with the substrate. Both of these will affect the enzyme's activity. So let's get back to important things to remember about enzymes. They are proteins. They are specific to the substrates that they act on. So amylase is not going to do the same thing as catalase or lactase or DNA polymerase. They do remain unchanged after reaction, so they're reusable. You can use them again and again. They work in a specific environment. So many enzymes have what's called an optimum pH or an optimum temperature where they function best. We'll see the rate of activity is going to be the highest there. And in general, enzymes work by lowering the activation energy of a reaction. Many times, biological reactions can occur without the presence of an enzyme. It just takes more energy to get there. So enzymes work by lowering that activation energy, making it easier to undergo the reaction and having more reaction activity within the cell. Synthesis is the process of using sunlight and carbon dioxide to make food in plants. We're going to get a little bit more specific and revise this just a little bit because we know not only plants do photosynthesis, but also algae and some bacteria. And it's not just food that they're making. So we're going to start talking about glucose, that organic compound. It's a carbohydrate, C6H12O6. So our basic equation for photosynthesis is sunlight plus carbon dioxide plus water make or yield, that's what that arrow means, glucose and oxygen. So plants are taking in these things, sunlight, carbon dioxide, and water, and then they are making glucose and oxygen during the process of photosynthesis. So if we start big at this tree and then we zoom into a leaf where the green stuff is, that's where photosynthesis is going to happen. We look on the outer layer of the leaf and we see different types of cells. These are actually the epidermal cells on the leaf. And then you might be able to see these little green dots inside these cells on the micrograph, and those are the chloroplasts. Chlorophyll is the pigment within chloroplasts that makes them green, and it's the chlorophyll that's actually absorbing the sunlight in the process of photosynthesis. So let's talk a little bit more about these ingredients. Sunlight is sunlight. Carbon dioxide can also be abbreviated as CO2, C for carbon, O for oxygen, meaning there's 2 atoms of oxygen and 1 atom of carbon in each carbon dioxide molecule. Water, of course, is H2O, and glucose is C6H12O6, and oxygen, of course, is O2. So this is our full equation for photosynthesis, not balanced. Sometimes you'll see the 6's in front of these molecules to make the balanced equation. What's really important is that the glucose and oxygen that are produced in photosynthesis are going to go directly into the process of cellular respiration, which cells use to make energy. And remember, plants don't just do photosynthesis, they also do cellular respiration. Cellular respiration takes place in the mitochondria. Photosynthesis takes place in the chloroplasts, but plant cells have both. As you can see here in our very simple plant cell diagram, here's our chloroplast and here are 2 mitochondria. One thing your teacher might ask you is to reverse this equation and see what it might look like, and it definitely looks very similar to the equation of cellular respiration because the inputs of one are the outputs of the other. All living things need energy, and one of the main ways they get energy is through a process called cellular respiration. Often when we say cellular respiration, we're actually referring to aerobic cellular respiration, meaning the process by which we use oxygen and glucose and then undergo lots of different reactions to get ATP, the cell's energy, and we produce the byproducts, carbon dioxide and water. If we think about this on an organismal level, this is happening across our body in trillions of cells, but the glucose we get from consuming, so we take in food, we digest it, and we get glucose from the food that we eat, and oxygen we inhale, and then throughout our cells, we're going to generate carbon dioxide as a byproduct. So we exhale that excess carbon dioxide, and then we exhale water vapor as well, and that comes from cellular respiration too. Now the ATP we don't really exhale, we use that in our cells for things like active transport and other processes that require energy. But cellular respiration takes place anywhere there's a mitochondria. So virtually all eukaryotic organisms that have mitochondria are going to use this type of energy pathway. Now even organisms that perform photosynthesis, like plants, will also have mitochondria because they'll take those products from photosynthesis and then they'll go into our cellular respiration reactions. Now if we look at our equation, our summary equation for cellular respiration, it's glucose and oxygen yield, water, carbon dioxide, and ATP. Again, this is the unbalanced version of that equation. And then if we look at the reverse, sort of, we see that photosynthesis, which we covered in yesterday's video, looks very similar to the cellular respiration equation because, in fact, the inputs of one are the outputs of another. Virtually. We're not going to talk about sunlight too much here. So we see that in cellular respiration, we need oxygen and glucose, which is produced from photosynthesis, and then as a byproduct, carbon dioxide and water are generated in cellular respiration. The energy that's generated in cellular respiration doesn't go back into photosynthesis. It's sunlight energy that's captured for photosynthesis, and then that process can feed cellular respiration. So very briefly, yesterday we talked about aerobic cellular respiration, where we go through all of the steps of glycolysis and then the Krebs cycle and then oxidative phosphorylation and the electron transport chain, if you got that far in the video. But the important thing to know is that oxygen is truly an important ingredient, but sometimes some organisms who are capable of performing respiration cannot do aerobic respiration because they don't have enough oxygen in the cell or in the mitochondria. Other times, other organisms can only perform anaerobic processes. Now, anaerobic, an means not or without, aerobic with air, so not with air, means without air or oxygen. Now, here's where it gets a little tricky, and sometimes teachers will call this the same thing. Anaerobic respiration is technically when we go through that electron transport chain at the end of our cellular respiration reactions, and there's something other than oxygen to accept the final electrons. Fermentation is an extension of glycolysis. So after glycolysis, we've generated a little ATP, then there's going to be some more reactions that the cell undergoes without going into the other steps of cellular respiration. So we're not going to do the Krebs cycle. It can be done without the presence of mitochondria. So all of those details are a little bit high level. So if you're just in regular biology or you're not quite ready to get in the weeds with that college level or AP level biology content, it's okay. Just be aware that aerobic cellular respiration involves oxygen and anaerobic respiration does not. Now, in comparison, one of the main things you want to remember is that compared to aerobic cellular respiration, which produces 36 ATP, fermentation only produces 2 ATP. Now I have a little bacteria on the screen because many bacteria can perform fermentation, other single cell organisms like yeast often do it, but we can also do it in our cells as well. Now there's different types of fermentation, which we'll get into. So in our muscle cells, for example, this is one type of fermentation here, which is called lactic acid fermentation based on one of the byproducts. So instead of having glucose and oxygen working together, we just have glucose. And at the end of these reactions, we are left with lactic acid and ATP. Now, we used to think that when our muscles get really sore in workouts, it's because of this lactic acid and because our body's been doing so much lactic acid fermentation. Scientists are now kind of debunking that idea and saying that the soreness is not really from lactic acid, it's more likely from little micro tears in your muscles. But lactic acid is a byproduct of lactic acid fermentation, and we do perform it in our muscle cells when we run low on oxygen. Now there's another type of fermentation, alcoholic fermentation, which can happen in yeast, it can happen in some bacteria. This process can also be completed without the presence of oxygen, and it produces carbon dioxide, ethanol, which is a form of alcohol, and ATP. Still just 2 ATP, but it has those other byproducts, carbon dioxide and ethanol. Now let's take a look at this chart comparing the products and what goes in and what goes out of each of these. So within cellular respiration, remember we need glucose and oxygen. That's what makes it an aerobic process. Aerobic process, not anaerobic. And then the products are 36 ATP molecules and carbon dioxide. Now, in our two types of fermentation we just talked about, lactic acid fermentation also requires glucose. Alcoholic fermentation also requires glucose. They are both anaerobic processes, meaning they occur without the presence of oxygen. They both produce 2 ATP. Lactic acid fermentation has also lactic acid as a byproduct, and alcohol fermentation produces ethanol and CO2 as byproducts. First, a little bit of vocabulary. Some students mix up chromosomes, chromatin, centromeres, sister chromatids, so we're going to go over that right now. First of all, this is a very bad illustration of a chromosome, and it's kind of representing a misconception that a lot of students have about what chromosomes actually are. The DNA in a chromosome isn't actually just one strand with, a couple dozen bases. In fact, it is millions of bases. Many chromosomes contain 50 million nucleotide base pairs, some even 300 million nucleotide base pairs. So what we're seeing here is a very bad representation of a chromosome. Remember, chromosomes are DNA that are tightly condensed and packed, wound up around specific proteins called histones. So let's talk a little bit about that organization and then we'll get into the vocab. So if we have our DNA molecule, we know that in the middle it's connected with base pairs, there's a backbone. When it starts to be wound up, it is wound around these protein complexes that can unwind to be transcribed or expressed. And when it prepares for cell division, it has to be very tightly wound into these structures called chromosomes. So that winding of the DNA around these proteins is going to be very important for us to organize the DNA in a way that allows it to be distributed evenly into the daughter cells. So when it is in the nucleus and it's not going through mitosis, it's not wound compactly into chromosome forms, we call it chromatin. Chromatin is just that complex of DNA and protein. Sometimes it's shown here, you can see it kind of tiny, it's sort of this like spaghetti-like substance. Now, when it prepares for cell division, the DNA, and you can kind of see it here, will condense into those chromosomes. In humans, we have 46, so it'll condense into those 46 individual chromosomes. Remember, these are long, long strands of DNA that are being packed tightly together. And before it goes through mitosis as well, the DNA has to duplicate. So each chromosome actually has 2 identical sister chromatids. They're the same on each side. And in mitosis, those chromatids are going to split apart. So let's talk about that vocab one more time. This whole thing is a chromosome. and you can see the individual chromosomes in the nucleus. But when it's not condensed into the chromosomes, we call it chromatin, or just that bundle of DNA and proteins is chromatin. The chromosome is attached at the center by a centromere, so centromere, center. And then each side of the chromosome has an identical sister chromatid. So one section here, that's one chromatid, and one section here, that's one chromatid. So one more time, chromosome, chromatid, centromere, and chromatin here in the center. Okay, we're going to be referencing these throughout today's reviews. I just want to make sure you had them all straight. We want to talk about the cell cycle, which is how a typical cell is going to go through its life. And so the main part of a cell's life cycle is called interphase, which involves stages G1, S, and G2, and that takes up the bulk of the cell's activities. So this interphase stage actually lasts a really long time. G1 is for growth, S is for synthesis of the DNA, so where the DNA is duplicated, and G2 is also for growth and preparation for division. After the cell divides, it can continue going through this cycle again and again, as long as the right signals are being processed within the cell. If we zoom in and talk about mitosis and cytokinesis specifically, we're going to look at the main stages of mitosis for how the cell is going to divide its genetic information so that its daughter cells have the exact same stuff as the parent cell. We can remember the stages of mitosis as PMAT, prophase, metaphase, anaphase, and telophase. And if we look on here, prophase involves the DNA condensing, our nuclear membrane dissolving, and our chromosomes sort of migrating to the center of the cell. Metaphase is going to involve those chromosomes lining up in the middle of the cell along the cellular equator, and the spindle fibers are going to start to attach to the centromeres of the chromosomes. In anaphase, those sister chromatids are pulled away, A for away, from each other, and those spindle fibers are going to pull them to opposite ends of the cell. In telophase, we start to get new nuclear membranes reforming, and then in cytokinesis, the cytoplasm is actually going to split, and we're going to have a cleavage, and we're going to get two daughter cells. The DNA is decondensing and going back into chromatin. All right, so again, a couple things to remember about mitosis. In a regular body cell, we're typically going to go from a diploid cell with just the regular amount of chromosomes, which is represented by 2N, to two daughter cells that are also diploid. So in mitosis, we go from diploid to diploid. We go from one cell to two cells, and they have identical genetic information as the parent cell. So the point of this is to make two identical daughters. Now, the DNA has to be duplicated in S phase before it can undergo mitosis, or else if we split the DNA in half without duplication, we would end up with half the DNA in the daughter cells, which is not what we want in the process of mitosis. That's for meiosis, which we'll talk about later on. So S phase has to happen during interphase, this longer stage of the cell cycle, in order for the DNA to be duplicated, and we can have enough in order to split it later down the road. Okay, so again, once we've duplicated that DNA in prophase, it's going to condense and get ready for cell division. Metaphase line up in the middle, anaphase will pull apart, telophase, we're going to get two new nuclear membranes starting to form, and then cytokinesis, we're going to get cleavage, and we're going to cut those cells in half. Okay, let's talk about the cell cycle now and these checkpoints that are regulated throughout a cell's life cycle. The cell cycle has multiple checkpoints or control points where we have these stop and go signals that are going to regulate how the cell cycle will continue. A lot of this is dependent on proteins and things called cyclins. And these cyclins are proteins that are going to go up and down in different concentrations in the cell depending on which stage of the cell cycle it's in. Just a few of some of the important checkpoints in the cell cycle are our G1 checkpoint, which will allow it to go through S phase, DNA replication, G2, and then mitosis and divide. But if it doesn't get the go-ahead at that checkpoint, then it can enter a G0 phase, which is a non-dividing state that a lot of body cells end up in. Another one here, at the M phase, is going to ensure that all the spindle fibers are properly attached to the centromeres of the chromosomes and they're all lined up in the right way. And if the cell passes that checkpoint, it receives the go-ahead signal, then it allows the cell to proceed into anaphase, separating those sister chromatids and pulling them to opposite ends of the cell. But if some of the fibers are not attached to the chromosomes in the right way, then the cell is going to receive a stop signal and it should not continue dividing. If we have any signaling pathways that are not functioning properly within the cell, especially related to some of these checkpoints, that's when the cell is going to divide when it shouldn't. We could have unequal distribution of chromosomes. We could have cells that ignore signals for apoptosis or programmed cell death, or ignoring that checkpoint during G1 to enter a non-dividing state. So remember that cancer is a loss of control of these checkpoints throughout the cell cycle. And usually that's because there's a malfunction in a gene that controls the proteins involved in these signaling pathways. A lot of cancer cells will continue dividing indefinitely in the lab if they're given all the nutrients they need, because they can just bypass the checkpoints whenever they want and continue to divide and divide. The phases of mitosis can be remembered as PMAT, or prophase, metaphase, anaphase, and telophase. And with meiosis, you might have heard that we go through PMAT twice, which is true, but we are going to add in some additional information today as we're talking about our review. Now, one of the main differences between meiosis and mitosis is the end goal. Mitosis's goal is to create two identical daughter cells. Meiosis, the end product, is 4 haploid daughter cells. And haploid means it has half the genetic information of the parent cell. Parent cell starts out as diploid, and then each of those daughter cells have half of the original information. This is useful for creating gametes or sex cells so that they can combine in fertilization to then create new diploid cells. One way I like to remember the difference so I don't get mixed up is thinking about mitosis is useful for growth and development and repair in larger multicellular organisms. So if I got a bump or a scratch on my toe, I would go through mitosis. In order to fix that, my cells would divide and create exact copies. But to generate me, I had to come from an egg cell and a sperm cell, and each of those was generated through meiosis. So me, meiosis, mito, mitosis. All right, so here are all the stages of mitosis together. We have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis, and then we do it all again. Prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So let's talk about what happens in each of these at a little bit closer level. So in meiosis 1, we have prophase 1 first, and our chromosomes condense and become visible, just like they do in prophase of mitosis. Our homologous chromosomes are going to pair up, and we're going to have a process called crossing over, where we have an exchange of genetic information between homologous chromosomes. This is really key in increasing genetic variation in organisms, but we have chromosome from one parent, chromosome from the other parent, and they're lining up together and they're exchanging genetic information through a synapsis. And after this crossing over occurs, we have a pretty unique combination of genes from both parents, and this is why no two siblings, unless they're monozygotic or identical twins, are going to have the exact same DNA. In metaphase 1, we're going to have our homologous chromosome pairs lining up at the cellular equator. And then instead of separating the sister chromatids, in anaphase 1 of meiosis, we have homologous chromosomes separated by those spindle fibers. And so at the end, we end up having full duplicated chromosomes still at the end of telophase 1. Then our cell splits, then it's time to do it all again. Remember, at this point, sister chromatids are still attached. We have to separate them in the next part of meiosis. So in meiosis 2, we have 2 rows here because we have 2 cells already, and remember, we're going to end up with 4 cells, so we have to undergo this division again. In prophase 2, our chromosomes will prepare again for this division, condensing if necessary. Our spindle fibers are going to form, and then in metaphase 2, we have our chromosomes lining up at the cell's equator. Notice that this part of meiosis looks a lot like mitosis. This part is more closely aligned to what we see in mitosis. So we have those chromosomes lining up just in one row this time on the cellular equator, and then in anaphase 2, sister chromatids are being pulled apart to opposite ends of the cell, and then in telophase 2, we have our genetic information fully separated, a nuclear envelope starts to form again, and then we're going to have cytokinesis when that actual split occurs, resulting in 4 daughter cells with half the genetic information of the original parent and different combinations of those genes as well. And here's the full process again, but I invite you to look in your textbook or any other diagrams your teachers provided to get a closer picture. Species have to be able to reproduce in order to pass on their traits and their genes and to survive. Some organisms reproduce asexually, and virtually all of their genes come from the same parent, and so they're copies of the parent organism. Other organisms reproduce sexually and have half of the genetic information come from 2 different parents. So 1/2 comes from one parent and 1/2 of the genetic information comes from another parent, and those 2 haploid cells are combined. Remember, in order to get those haploid cells or gametes or sex cells, for sexual reproduction, we have to go through meiosis first. The process of meiosis results in half the genetic information in the daughter cells, and in humans, This happens during the production of eggs or sperm. We can get lots of genetic variation through meiosis, through independent assortment of chromosomes, crossing over of chromosomes during prophase 1 of meiosis, and then of course the random fertilization of one egg cell with any one sperm cell. Remember, mutation is an important source of genetic variation, but these 3 reasons on this slide are very, very important as well. The process of meiosis is important in sexual reproduction because it provides genetic variation of offspring, and genetic diversity is important for the survival of a species. So in order to get that egg and that sperm, we have to go through the process of meiosis first. Then those two can undergo fertilization to produce a zygote, which is a fertilized egg cell and now a diploid cell. So let's put this all together. In humans, both a biological male and a biological female will need to undergo meiosis in order to produce eggs and sperm. Then one egg cell and one sperm cell will combine in fertilization. After that, that fertilized egg will divide and make copies of itself through mitosis as the embryo starts to grow. Mitosis will continue until the cells start to differentiate and become all the different important parts of the growing embryo. Remember, differentiation is how we get so many different types of cells from the same DNA. Even though we have the same DNA in every single one of our cells, different parts of that DNA are turned on or off in order to produce the proteins to make the differences in all of our cells. Human reproduction and development are influenced by a lot of different factors, including hormones, the environment, and gene expression. In both males and females, our reproductive cycle is regulated by the cycling of different hormones throughout the body, including testosterone, estrogen, and progesterone. The cycling of hormones during the female menstrual cycle, as shown here, is going to involve a series of natural changes in hormone levels in order to produce the physiological effects in the uterus and the ovaries that are going to make pregnancy possible. Let's take a look at our gonads, or our sex organs, in human males and females. We have gametes, or our egg cells produced in the ovaries, and then those eggs will leave through the fallopian tubes seen here. Once a fertilized egg implants in the lining of the uterus is going to provide an environment that's going to support the development of the embryo and the fetus. The placenta, which is connected to the developing embryo through the umbilical cord, is going to provide essential nutrients and the materials for the developing fetus. In males, the sperm are produced in the testes, as seen here, and both sperm and urine are going to pass through the urethra in the male reproductive system. During embryonic growth in human pregnancy, the development of important essential organs occurs early during the developmental stages. Here again, we see another impact of environment on genes, because if the mother is exposing herself or the fetus to negative environmental conditions like alcohol or tobacco smoke, that could have a very detrimental effect on the growing embryo. That's why it's important to avoid the use of alcohol, drugs, and tobacco during pregnancy.
Guys, so we've studied mitosis and meiosis. Now we're going to focus on comparing the two processes together. Now I know they may get a little confusing, they even sound the same, but we're going to review their differences and similarities one more time. Alright, so for starters, meiosis, of course, we are making sex cells like our sperm and our egg in humans. For mitosis, we're just making regular old body cells. We call these somatic cells. Now these cells will all have the exact same DNA, whereas these cells are going to have slightly different genetic information and different amounts. But let's get to the nitty-gritty. So remember, the process of meiosis is a little bit more complicated than the process of mitosis. If we started out with the same genetic information, so if this pretend organism we're looking at had 2 chromosomes, if it goes through mitosis, remember, it goes through prophase, the chromosomes condense, it's going to line up in the middle with metaphase. In anaphase, those sister chromatids are pulled apart, and then in telophase, we're going to have chromosomes decondensing. We'll start doing the process of cell separation, the new nuclear membranes are forming, and we're almost ready for cytokinesis, and we're going to have 2 identical daughter cells that look just like the parents. You can see down here, they're the exact same as what we started with. In meiosis, however, if we're starting with a parent cell that looks like this, we're going to end up with a daughter cell with half the genetic information that will be genetically different from the parent cell. We're going to go through PMAT again, prophase, metaphase, anaphase, telophase, but we'll do it twice. Now remember, before both these things can start, how do we get from here to here and here to here? Remember, the DNA is replicating in interphase during that S phase. So the DNA is going to copy itself over to make these beautiful butterfly-looking chromosomes so that it can later on separate. That's how we don't lose any DNA in the process. If the DNA didn't replicate it, we would be like no DNA at all by the time the process was done a couple times.
So let's take it way back. Mendel was the person that deduced that genes come in pairs and are inherited as distinct units from one parent. He tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. He also recognized the mathematical patterns of inheritance from one generation to the next, and he created laws of heredity. Mendel did a lot of great work in his experiments, and they took about 8 years to complete. He published his work in 1865, but unfortunately he wasn't really recognized or thought of as important until after his time. We know that he worked with over 10,000 different pea plants during his experiments, and he took meticulous results, recording down all of his data by hand. All right, and if you have to memorize Mendel's laws, an easy way to remember them is pretend that at the monastery, Gregor Mendel, who was a monk, had a nickname named Sid. I don't know why, it's just part of the story, but Sid will help you remember the 3 main laws that he came up with: segregation, independent assortment, and dominance. A gene or genes are pieces of DNA that determine your traits. An allele is just a different form of a gene. If we're writing out genetics problems, this might be written as a big B or a little b, for example. A recessive gene can be masked by a dominant one, and a dominant allele can mask a recessive one. Often dominant alleles are represented by capital letters. A genotype is the actual combination of alleles that somebody has. A phenotype are the physical characteristics that come from that genotype. So for example, your physical characteristic could be tall. That's your phenotype. Homozygous is when we have an allele pair that has 2 of the same allele. So for example, a little a or a big A, big A. Heterozygous is when we have 2 different alleles together in one genotypic pair. For example, big A, little a. So some basic review for Mendelian inheritance. This is our classical dominant recessive type of inheritance. Remember that genes are often represented by letters, and different versions of genes could be capital or lowercase letters. So here we see the trait for this axolotl. The color of their skin is either going to be pigmented, so this dark color, or without pigment, so this albino axolotl. And to represent these different versions of these traits, we have letters that are going to be different alleles. So the pigment allele will be a big A, and the albino allele will be a little a. Now, each organism has a genotype. These organisms have 2 genes for a particular trait. So this albino axolotl is homozygous recessive, meaning it has 2 little a's. So little a would be this axolotl's genotype, but its phenotype, its physical characteristic for the trait, is albino. That's the color. So remember, phenotype, physical characteristic. Genotype is the actual combination of alleles or genes. Now, this pigmented axolotl could be one of 2 genotypes. It could either be big A, homozygous dominant, or it could be big A, little a, because this big A is dominant over the recessive allele, and it's going to mask or cover up the effects of this recessive gene. So when we see a pigmented axolotl, it could be either one of these genotypes, but both of these genotypes will give us the pigmented phenotype. All right, so let's take a look at a practice problem involving these and our tool, the Punnett square. So let's say we have 2 pigmented axolotls that are both heterozygous, meaning they have one big A and one little a, and they are going to mate. We can use things like a Punnett square to see what the chances are of their offspring either being albino or having pigment. So in this case, to set up a Punnett square, we would take one parent and put it on the top of the Punnett square, separating out their alleles or their letters. We would take the other parent and separate out their alleles or letters as well. And then what we do is we drop down all our letters and we fill in the Punnett square. So this big A gets dropped down here and here. This little a gets dropped down here and here. And this big A gets dragged over here. And this little a gets dragged over here. So now we have the results of our Punnett square. We can see that there's a 25% chance of having offspring that will have a homozygous dominant genotype, meaning big A, a 50% chance of having offspring that will have the heterozygous genotype, so big A, little a, and a 25% chance or 1 in 4 chance of having little a, or homozygous recessive. Now, that's a lot of words. What does that mean? So these 3 combinations here, big A, or big A, little a, that would all mean this axolotl has pigment or is a dark color because A is dominant over little a. So we have 2 copies here. It doesn't really matter. Here we have 1 copy of the dominant trait, so it's going to be dominant over the albino trait. An axolotl with little a, though, that genotype means it doesn't have any pigment because it has 2 copies of the recessive allele. In humans, most human traits are very complicated genetics-wise. In fact, most human traits are polygenic or polygenic, meaning they are controlled by many different genes interacting together. There's very few classic Mendelian inheritance instances that show up in humans. So some of these are actually genetic disorders like cystic fibrosis, sickle cell anemia, PKU, and we'll talk more about genetic disorders when we get to chromosomes too, which is tomorrow's video. But I just wanted to mention that a lot of examples that we might see or use in biology classes are actually incomplete versions of what's really happening with human genes, because many human traits are caused by combinations of up to thousands of different genes interacting together. And the interactions of different genes can cause differences in phenotypes, and we have a spectrum or a range of different phenotypes in many instances. For example, hair color, skin color, it's not just a yellow pea or a green pea. It's much more complex than that. And then of course we have epigenetics too, where we have different factors that are controlling which genes are turned on and off in which environment. So it does get very complex. But today what we're going to talk about are patterns of inheritance where we don't just have one single dominant allele and one single recessive allele. We can have things such as incomplete dominant. This is when we have a blending of traits. So there may be two different alleles here. For example, the classic one is something like a red flower and a white flower. We'll pretend this is white on the screen. And then if they are crossed or bred, they would generate pink flowers. So let's see how this works. So let's say that in this incomplete dominant scenario, we're going to represent red flowers with big R, white flowers with big W, big W. Notice how these are two capital letters for both alleles. And we're using different letters too instead of R's. That would get really confusing if they were both big R's here. So we set the Punnett square cross up like we would do any classic Punnett square, and we see that our combinations in this case would only be big R, big W, RW, 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 RW. And so what that results in is a phenotype that's a blend of the two parent traits. So RW would actually give us pink, that intermediate characteristic, instead of one or the other, which is really interesting. Codominance is when we have a representation of both traits in the organism. So we're not meeting in the middle, but instead we have both traits show up at some level. Roan cattle are a very common example of this. But with flowers, again, to do it very simply, let's say we have purple flowers and pink flowers. Instead of getting a blend of that purple and pink, we would get some petals that are purple and some petals that are pink, or some spots in the petals that are completely purple and some spots in the petals that are completely pink. So let's see how this works with the Punnett square. In this case too, we use capital letters, two different letters to represent the two different traits. Big P for purple, big B for pink. We cross these two. What we end up getting is, just like we saw in the previous example, these heterozygous offspring that are a combination of both the purple allele and the pink allele. And so what that gives us are flowers that are not a blend of the two colors, but instead show both traits. Blood types we'll see as an example for codominance very frequently. Now, blood types are very interesting. They're controlled by several different alleles. So we have something called multiple alleles that are a possibility here. It's not just A or B. We can have the allele for blood type A, the allele for blood type B, or the allele for blood type O. So that's three different alleles. And then remember, a genotype is two alleles together. So we have lots of different possible combinations here. And AB blood is actually a codominant genotype. So if someone has the allele for type A and the allele for type B, they will represent antigens for both blood type A and blood type B. And then we can talk about really interesting patterns that occur because type O is actually recessive, but A and B are codominant. So that's a good example of codominance. And then of course there's the Rh factor and lots of different things we can talk about with the different blood types and who's a donor and whatnot. But let's move ahead now to talk about sex-linked traits, which is another different type of inheritance that we see in humans. So these are traits that have alleles that are located on sex chromosomes. So in a human karyotype, there are 23 pairs, so 46 total chromosomes, and 2 of those are sex chromosomes. In biologic females, it's an X and an X. In biological males, it's an X and a Y. If something is an X-linked trait, that means it appears on the X chromosome. Generally, there's going to be more X-linked traits that we'll talk about just because the X chromosome is much bigger and there's more space for traits. Colorblindness is a very common X-linked recessive trait, as is hemophilia. That is also X-linked recessive. And Y-linked traits, there's fewer of them that we'll study and talk about just because the Y chromosome is smaller. And many Y-linked traits actually lead to sterility. So if someone had one of those Y-linked traits, they could not pass it on to their offspring because if someone is sterile, they cannot have offspring. All right, so let's just take a look really quick. Remember, females have the XX chromosome pair and males have an X and a Y. If we look at a human karyotype, so 1 through 22, these are all the autosomes or the non-sex chromosomes that humans have, and then the 2 sex chromosomes are either XX or XY, and so this is a karyotype of a human male. So let's take a look at the inheritance for color blindness. So the allele for normal vision is dominant, and the allele for color blindness, or someone who is affected with this color blindness trait, is recessive. So we'll show that with a little h. Here we show two normal individuals. Neither of them carry any of the genes for color blindness. In this case, this female is a carrier here. She is still normal though, because she has one dominant allele, that big H, and that can mask or cover up the little h. So she is still fine. She has normal vision. She's not colorblind. The male here is normal as well. Now in this case, the female is colorblind. She has two of those alleles for colorblindness. They're both carried on the X chromosome. The male is also normal. And here we have two colorblind individuals. We have a colorblind female and a colorblind male. But notice with the male, he only needs one copy of that recessive allele in order to be colorblind, because he only has one X chromosome. So there's no dominant allele present to be able to mask that other allele, and so he will be colorblind with that one copy of the affected allele. And here we have a case with a female who is normal. Her alleles are unaffected and the male is colorblind. All right, so how do we set up a Punnett square for a sex-linked cross? It's not overly complicated. We just have to make sure we carry the X's and the H's or whatever letter we're using for the trait together, and we don't separate those, but we are going to separate each individual sex chromosome. So if we were to cross these two parents, we would set up the Punnett square like this, and we make sure we write the letters as like an exponent, like it shows in their genotypes up at the top. And then just like a regular Punnett square, we'll carry down the letters, bring them across to see the possible combinations for the offspring. So in this case, we could have, out of all the possible offspring, there's a 50% chance that this couple will produce females who are carriers for color blindness but not have color blindness themselves, and a 50% chance that they will have offspring who are males who are normal. They don't carry any of the colorblind alleles. So remember, females can be carriers of sex-linked traits because they have two X chromosomes, and technically in biology, a carrier is someone who does not display the trait, but they could pass it on to their offspring. So in this case, a carrier for color blindness would look like this, and males technically cannot be carriers of sex-linked traits. So obviously someone who has the trait could pass it on to their offspring, but a carrier, if we're going by that strict definition, males can have conditions that are sex-linked, but they're not carriers technically. Genes can be influenced by the environment. For example, in identical twin studies, we can see that identical twins separated at birth can have different heights and weights after 20 years. Most likely the cause of these differences is that each twin was provided a different diet and had different physical activities throughout their lifetimes, because an environment does play a role in influencing individuals. So let's look at a karyotype. Now remember, a karyotype is just an image of all the chromosomes in the nucleus of an organism's any one cell. So in humans, we have 46 total chromosomes, or 23 pairs. So remember that the first 22 pairs, these are called autosomes, and the two that are left, these are our sex chromosomes. If you're biologically female, you have two X sex chromosomes. If you're biologically male, your sex chromosomes are X and Y. Now we do see some differences in certain genetic disorders where we can have different combinations of these, but we'll get into that when we start talking about genetic differences. So how do we get a karyotype of a baby that's not born yet? Well, we can use something called amniocentesis. Amniocentesis, or an amniotic fluid test, is a medical procedure used in prenatal diagnosis of genetic disorders and fetal abnormalities. So what happens is a small amount of amniotic fluid, which is here, is going to contain some fetal tissues, and it's going to be extracted from the amniotic sac with a large needle. And once we take it out, the DNA can be examined for genetic abnormalities, or we can simply see the biological sex of the individual. Now, not all women choose to do this because there is a small risk involved in the procedure of amniocentesis, but if you elect to do it, or if you're high-risk pregnancy, some women will be able to see a karyotype of their child before they're born. Now remember, a normal human karyotype has 23 pairs of chromosomes, including the sex chromosomes here. This individual has all 23 pairs, one X and one Y. If you look closely, you can see this individual is a biological male. So when we look at our karyotype, we're going to identify and evaluate the size and the shape and the number of chromosomes in our sample body of cells. Some problems that we could see would be duplicates of chromosomes where there shouldn't be, or deletions of certain arms of chromosomes, or translocations of chromosomes. One difference we could see right away is if there was a third chromosome at the 21st position. Now, all the chromosomes aren't arranged like this. Scientists have to make sure that they are arranged after the picture is taken into the correct order for the correct chromosomes. But once they are arranged, we can look and determine if there's any genetic abnormalities. For example, At the 21st location, if there's a third chromosome, we would have what's called trisomy 21, also known as Down syndrome. Trisomy just means 3 copies of a chromosome. Now, chromosomes aren't everything and genes aren't everything. The environment has a huge influence on phenotype or on the resulting traits of an individual. Some scientists estimate up to 75% of who you are and how your traits operate is due to environmental factors. We're learning so much more about epigenetics and how different things can turn genes on and off in different ways. So environmental factors can influence gene expression and lead to phenotypic plasticity. This is when individuals with the same genotype have differences in the phenotype that they express because of environmental conditions. For example, there's some mammals like rabbits that can exhibit different fur colors and different temperatures. Hydrangeas are another popular example of this. The petals of certain hydrangeas can change color depending on the pH of the soil that they're planted in. So, for example, here we see that blue flowers are going to appear when the soil is highly acidic, and pink flowers are going to appear when there's a neutral or slightly acidic soil. Many reptiles, such as crocodiles and turtles, have sex determination based on temperature because sex determination in these animals is highly dependent on hormones, and these hormones can be changed by the activity levels of different enzymes depending on a specific temperature. So we can see an entire brood of turtles hatching to be one sex because they were all incubated at a particular temperature. We know that some genetic differences in individuals are due to single allele or single gene changes, and others are due to changes in entire chromosomes. A lot of times this happens because of non-disjunction or when a chromosome doesn't separate properly during meiosis. A lot of genetic disorders we call autosomal because they're occurring on any chromosome that's not a sex chromosome. So all the chromosomes except for sex chromosomes, remember, are autosomes. And so autosomal conditions are caused by mutations on chromosomes that are not sex chromosomes. You might have heard of Huntington's disease as an example of an autosomal condition or an autosomal dominant genetic disorder, which often doesn't appear until later in life, so people can pass it on to their offspring without even knowing that they have it. And then there's lots of autosomal recessive disorders like Tay-Sachs disease, cystic fibrosis, PKU, sickle cell anemia. Chromosomal conditions are usually caused by non-disjunction, which means when those chromosomes are supposed to separate, especially during anaphase 2 of meiosis, we're getting an extra chromosome ending up in the sex cell. So things like trisomy 21, Turner syndrome, Klinefelter's, poly-X, Jacob syndrome, these are all examples of chromosomal conditions. Now, as a reminder, DNA, one of our most important nucleic acids, is found in the nucleus of eukaryotic organisms or in the nucleoid of prokaryotic organisms. DNA stands for deoxyribonucleic acid. We're also going to talk about ribonucleic acid or RNA. So again, one nucleotide contains a phosphate group, our deoxyribose sugar, as well as our base, which can be A, T, G, or C in DNA. Here again, we're looking at many nucleotides put together. Our phosphate here, deoxyribose sugar, and our base. Now, importantly, when we look at this, our adenine and guanine are a little bit different from cytosine and thymine. Adenine and guanine are purines, meaning they have a double ring structure when we look up close. Pyridamines, which contain thymine and cytosine as well as uracil and RNA, have a single ring structure. Now, adenine and thymine are pairing together with 2 hydrogen bonds. Cytosine and guanine pair together with 3 hydrogen bonds. But these are easily broken during DNA replication or transcription. This is something you'll absolutely have to memorize for any biology class. So A always pairs with T, G always pairs with C when we're talking about DNA molecules. Some people like to remember this as apple tree good cookie or all teachers can go if you do it backwards. Whatever mnemonic that you have to remember this, make sure that A's and T's and G's and C's always go together. Another one that I've heard before that is helpful is that capital A's and T's are written with all straight lines and capital G's and C's have those curvy shapes, which is another way to remember which ones go together. Here are purines and pyridamines up close. You can see that adenine and guanine have 2 rings. Pyridamines have 1. A great way to remember this is pure as gold, cut the pie. This is a mnemonic to help us remember that our purines, pure as gold, are adenine and guanine, as seen as the as gold, A and G here. And our pyridamines are cut the pie, C and T are in the word cut, pie for pyridamines, PY. Hopefully that'll be helpful. Now let's talk a little bit about RNA. Remember, RNA is single-stranded. It's not going to have that double helix that DNA does. Both RNA and DNA do have our 3 components in our nucleotide, a sugar, a phosphate group, and a nitrogenous base. And these are going to form units connected by covalent bonds.
with 5' and 3' ends. But when we look at the sugar in DNA, that's going to be a deoxyribose sugar, hence the name, and RNA is going to have a ribose sugar. These are both pentose sugars with 5 carbons, but they're a little bit different. DNA is going to contain thymine, whereas RNA contains uracil. So instead of having a thymine, RNA is going to have that U. DNA will always have T's. DNA is usually double-stranded unless we're dealing with certain situations, and RNA is usually going to be single-stranded. DNA doesn't really leave the nucleus in eukaryotic organisms. RNA will leave the nucleus as mRNA or rRNA or different types of RNA for protein synthesis and other functions within the cell. Now, one nucleic acid we don't want to forget is ATP. Adenosine triphosphate, you might remember from studying cellular respiration in biology. ATP is also a nucleic acid, and you notice it has a very similar structure to DNA. We have phosphates, we have sugar, and we have our base. But unlike DNA or RNA, ATP has 3 phosphate groups attached to the sugar, and this is also a ribose sugar. And energy is formed from ATP when these phosphate groups are broken, and we get adenosine diphosphate instead of triphosphate. Chromatin, or the DNA that is in the nucleus in a eukaryotic organism, is typically wrapped around proteins called histones, which then group into something called a nucleosome. So we have lots of DNA-based pairs wrapped around core of histone proteins, and then these nucleosomes themselves are organized into different structures so that we can finally package it into chromosomes if necessary. Talk a little bit about DNA replication. Remember, it occurs during the S phase of our cell cycle to allow daughter cells to have an exact copy of the DNA after they go through cell division. If they didn't duplicate before they went through cell division, we would have half the DNA each time the cell divided, and soon almost no DNA at all. Now, with the help of some different enzymes, the DNA molecule is unzipped or opened up, and then we have 2 strands that we can add new nucleotides to. We'll have one old side of the DNA and then one new side in each of our 2 new DNA molecules. So remember, it's split open by our enzyme helicase, and then we'll add new nucleotides to each of those open sides, ending with 2 new molecules or 2 new DNA strands, each half old, each half new. This is why we call it half conservative, semi-conservative. Some of our important enzymes that we're going to talk about are helicase, polymerase, ligase, and topoisomerase, which is a mouthful. It's an enzyme that helps stabilize the DNA during the unwinding, so we don't have any supercoiling or tight twisting and coiling of our DNA. So it's a little bit ahead of where we're actually being unwound. Helicase I don't have depicted here, but this is going to be our unzipping or our molecular scissors that'll actually unzip our DNA. Polymerase is going to add to our template strand by building the new strand, so adding the new nucleotide sequences on or as a complement to the template strand. And then ligase is going to covalently connect sections of DNA that are being synthesized in fragments. So our DNA ligase is important here, especially when we get to our lagging strand or our Okazaki fragments. Now, remember that replication is a semi-conservative process, so one strand serves as a template for the new complementary strand. Each strand is anti-parallel, which means the 5' to 3' direction of one strand runs counter to the other 5' to 3' direction of the other strand. So if we have a 5' on this end, we would have a 3' on this end. And remember, those primes actually refer to the carbons in the sugar in that nucleotide. Now, DNA is found in the nucleus of a cell, and the DNA is usually in chromatin form, meaning it's unwound or decondensed, but when a cell is preparing for cell division, it'll condense into these chromosomes. So if we look here, we kind of see the organization of DNA. Our DNA, again, is wound into a double helix, and it's very, very long. We have over 20,000 genes in the body, and this long strand of DNA is wound around special proteins, and then that is all condensed up into a single chromosome. But each section of DNA that codes for a specific trait or provides instructions for a particular protein is called a gene. Now, not every single part of our DNA codes for a protein. Some parts of our DNA are there to regulate what parts of the DNA are turned on or off. Some we don't know what they do, and some, again, may be relics of viruses from the past. Sometimes chromosomes are represented like this, with each band or segment representing a particular gene or set of genes. We say DNA gives us our traits. So how do we get from a strand of DNA all the way to an actual trait, like the color of our eyes? Well, it all depends on proteins. And so protein synthesis is the process of how the message of DNA is transferred
transcribed and then translated into proteins. So synthesis just means making. So if you're trying to remember what protein synthesis is, just think about how we get proteins, how we make proteins from DNA. Let's see how this works. So remember, the DNA is in the nucleus of eukaryotic organisms. So we have our DNA strand, it's going to be split apart, and it's going to start to be copied as mRNA. And so this mRNA strand will build, basing itself off of part of that DNA strand. There's a couple of differences between mRNA and DNA, and one of those things is that instead of T's, RNA has U's. So when we pair with A, we're going to match it up with a U instead of a T in mRNA. RNA is also single-stranded, so once it's done copying the DNA, it'll leave the nucleus, the DNA will go back together, and then the mRNA will reach the ribosome where we can do the rest of the work, the translation part, where we're going to get our tRNAs, which are another type of RNA, are going to match up with the message on the mRNA and bring over these little molecules called amino acids. So glue here, that's one amino acid. And as these tRNAs match up with the code on the mRNA, we're going to bring more and more amino acids over, and they will be linked up by bonds here. These are peptide bonds, and we will start to form a protein. And once we have our amino acids linked together, we have our polypeptide or our many peptide chain, and that will result in our protein, which will do the job for the cell that we need it to do. It gets very long and it folds up in a specific shape, so it becomes a functional protein, like an enzyme, for example, or a pigment protein that'll color our eyes. So again, some differences between DNA and RNA. DNA is double-stranded. Think of that D for double. RNA is single-stranded. DNA has actually a different sugar than RNA. It's a deoxyribose sugar. RNA has a ribose sugar. And then, of course, DNA has T's, where RNA has a U as one of its bases. So let's practice here. If you want to pause the video and write down the mRNA sequence you think would go with this DNA strand, go ahead. I'll give you a second. And here it is. This is the RNA sequence that would come from this DNA strand. So now we have a message in our mRNA that can be taken out into translation. Now, if you confuse transcription and translation, remember transcription, we're changing from one type of nucleic acid, a deoxyribonucleic acid, into another type of nucleic acid, a ribonucleic acid. We're transcribing it into a message. So thinking about you writing down a message that you're going to pass to your friend, that's transcription. Translation, on the other hand, if you're translating something from English to French, you're changing languages, and that's what we're doing here in the cell as well. We're changing molecular language. And so we're going from a nucleic acid to an amino acid chain or a protein. So if you want to remember which one's transcription and translation, think about which one's writing down the message, that's DNA to mRNA, and which one is getting us our chain of amino acids, or we're changing our language. We're going from a nucleic acid to a protein, so that's translation. Now, you might be asked to read something like this called a codon chart or an amino acid table, where we're given all these letters here and it tells us which amino acids go with which three groups or codons of RNA. So for example, if our codon was UCA, we would start here in the center with the big letter U, and then we'd find the C in the next level, and then we would go to the outermost layer, A. Okay, so that's serine, or SER. This is an abbreviation for an amino acid. Let's try another one. How about AAA? So we start here in the center, A, and then we look here to see which one this touches. A, that's lysine, that's another amino acid. So every codon matches with a specific amino acid. Now there's other types of codon charts as well. You may see one that looks like this. You have to read them a little bit differently, but sometimes they're broken down very helpfully. So biotechnology can mean a lot of different things, but mostly when we say biotechnology in biology, we're referring to the modification of living things to create products for humans that are useful to us. Very commonly, these are things that are used in medicine or healthcare. You might have heard of the CRISPR-Cas9 technologies and how all of a sudden we have sickle cell treatments on the market now because of this biotechnology. But with any kind of rapidly evolving technology, there are going to be questions that come up. And so one of the things you might hear talked about in your classes is the ethics of biotechnology, or the ideas behind what we should do or we shouldn't do, and ethics aren't always black and white, so it's really good to talk about these things and discuss pros and cons and societal implications to any new technology that arises. So for example, we can now grow meat in a lab that we've taken living cells from cow muscle tissue. We have mosquitoes that we can release into populations that have been modified to be sterile or to produce sterile offspring when they mate with living mosquitoes in a population. This can reduce the spread of disease, but maybe this could have unforeseen effects on non-natural populations. There's also so many easily available at-home genetic testing kits that have a huge range of implications for society and the general public. We can select embryos with specific traits now as they're being used in IVF, and there are even de-extinction projects that are happening right now to bring extinct animals back from extinction by using really incredible biotechnology tools. So beyond all of these big picture ideas, we want to get into how we're actually doing this. What are we doing to the DNA? So there's some very important techniques we're going to cover, and there's others. There are many more out there, like DNA sequencing, that we aren't going to get into here, but let's just start with PCR. So the main idea of PCR is making lots of copies of DNA from a small sample. With PCR, we can make a lot of copies in a relatively short amount of time. What you need to know about PCR is that there's special machines that you can put your DNA sample after preparing it into, and then those machines go through cycles to do these three things. First, it heats up the DNA, which denatures it or separates it into two different strands. Then we're going to cool a little bit to 55 degrees Celsius, and primers, which have been added to our DNA sample, which are these short fragments of DNA, will be able to anneal or connect to the separated DNA fragments. From there, we're going to heat it up a little bit more so that we have more of an affinity for elongation. More nucleotides, which are floating in our mix, can attach to the open fragments of our DNA. So after one of these cycles, we're going to get two new strands of DNA. Then it goes through the same process again. They're denatured or separated. We have our annealing process, and then we have our elongation. And you see how each time we're able to double the amount of DNA. So PCR just stands for polymerase chain reaction, and we'll do this over and over and over again until we get enough copies of our DNA. After that, we can use this DNA to do a multitude of different things. One very popular use is getting a DNA fingerprint or doing gel electrophoresis. The first step in gel electrophoresis, or getting a DNA fingerprint, is going to be cutting that DNA with restriction enzymes. Restriction enzymes are special enzymes that cut DNA at specific recognition sites that are usually only a few base pairs in length. Sometimes these are palindromic, meaning they read the same forwards and backwards. But after that cut is made, we'll get what's called sticky or blunt ends at the edges of our DNA. And a sticky end is when we have bases that are unpaired after a cut. So let's see what that means. So we'll have our sequence of DNA. The restriction enzyme will recognize a specific sequence and cut along that site to give us two separate fragments of DNA. For example, BAMH1 cuts our DNA between the two Gs in this sequence whenever it encounters it along the strand. After we've prepared our DNA by cutting it with restriction enzymes, then we can put it in a gel. Our gel is a matrix-like material with tiny little holes in it that's going to allow our DNA to travel through it. Usually we put a marker or a sample of DNA that we know the exact fragment lengths of in the very first well, which is at the top of our gel. And then the other DNA samples that we're analyzing are going to be loaded into the next wells, usually with a dye, and we're doing this with a micropipette. Once we've loaded all of our samples in, what we'll do is we'll attach electrodes. So we're running charges. Up here, this is a negative charge, and down here, this is a positive charge. One very important thing to remember about the properties of DNA is that DNA is negatively charged. So if we run an electric current through this gel, DNA will start to travel towards the positive end. But remember, in each of these samples, we've chopped our DNA up. So as it travels down towards the positive end of our gel, we're going to see the DNA start to separate because each of these fragments is actually a different length. Our marker here is used for estimating the size of our DNA, but one thing we know for sure is that as the DNA travels down the gel, the longer fragments, or the larger ones, are going to stay closer to the top or closer to the wells, and the smaller fragments are going to be able to travel farther down the gel. So when the electric current is applied, the negatively charged DNA is going to move towards the positive end of the gel. But in each sample, each of these fragments are going to be different lengths, so we'll see different bands for the separation of the DNA molecule. One way I like to remember this is that if you have a race between one really, really large, probably out of shape person and one really, really tiny, pretty nimble, fit person, the person who is smaller is going to be able to run faster and win the race. And so that is what this DNA is doing. They're able to get through all of these holes in the gel faster because it's a smaller fragment and a smaller piece. You should be able to know the closer this is to where the wells are, the larger it is, the closer our DNA is to the positive end of our gel, the smaller it is. So now that we know we can separate DNA based on size, what can this help us with? Gel electrophoresis has a lot of purposes, from DNA analysis at a crime scene, paternity testing, looking at how closely two organisms are related, evolutionarily. But let's back it up to a crime scene. If we have, for example, our evidence from the crime scene, which is DNA, and we want to compare it to our suspects that we have in our case, what we can do is look at the banding pattern or the bands from the evidence DNA and compare that to the bands of the suspects. Based on these banding patterns, we can see that suspect 3 is a really good match with our evidence, and we might have just solved the crime. We'll also use restriction enzymes and gel electrophoresis when we're doing genetic transformation. And what we're doing with genetic transformation is inserting a gene of one organism into another organism so that it can display a new trait. Because of the central dogma, we're able to do this because virtually all organisms use the same universal genetic code. A lot of times we'll do this with bacteria and plasmids. But what is a plasmid? A plasmid is a circular double-stranded piece of DNA that's usually found in bacteria. It's extrachromosomal, meaning it doesn't belong inside the bacteria's chromosome, but bacteria will express or produce proteins from the DNA in these plasmids, and they'll replicate the plasmids whenever that bacteria divides as well. We use genetic engineering in a lot of fields, including agriculture, for example, inserting genes for pest resistance or fungal resistance into certain plants, in bioremediation, where we've genetically engineered bacteria to uptake oil at oil spills, or even in gene therapy in medicine. We've even used bacteria to produce insulin for humans before. When we talk about natural selection, we're talking about it as a mechanism of evolution. Natural selection is one of the major mechanisms of how organisms evolve on Earth. And evolution we can define as the change in genetic makeup of a population over time. Small changes can accumulate into massive ones over many millions of years and millions of generations. And natural selection explains how that can happen in different environments. When we talk about how an organism is evolutionarily fit, that is not about being strong or being big and bulky or fast. It's about how well they survive, reproduce, and pass on their traits to their offspring. One big deal to talk about in natural selection is the environment and how the environment can change and act as a selective mechanism on populations. And we'll get into some examples of that soon. Another key term that we're going to be talking about is adaptation. We have one definition here on screen. Another way to describe adaptation is a trait that increases fitness. So a trait that's going to help an organism survive and reproduce and pass their traits on to the next generation. Charles Darwin, as you might have heard before, is our father of modern evolutionary theory. His theory of natural selection was supported by evidence from many different scientific disciplines. He was an English naturalist, and he went to the Galapagos Islands, made a lot of observations, did a lot of critical studies, and his theories and his ideas behind natural selection really became the foundation of modern evolutionary studies. So these are the key components behind his ideas in his theory of natural selection. First, species can vary. So within a population, we can have individuals with different versions of traits. We know this when we were studying genetics. We saw there are different phenotypes within one particular population for a particular combination of genes. There is also the idea that species can change over time, and this is in relation to the populations. And last, there is a struggle for existence. So you might have heard the term before, survival of the fittest. And this just means that there's a competition for resources like space or food or light within a particular environment, and the individuals with the more favorable variations of a trait, so the best phenotypes for that environment, are more likely to survive and then produce more offspring and passing their favorable traits on to the future generation. And what else is important? We need a diverse gene pool for a population in order to survive and not go extinct. Environmental conditions can change. Something that is favorable in one environment could not be favorable in the future because of the environment changing. And we'll go over an example of this a little bit later in the video. This is a very important distinction I want you to make too. Individuals do not evolve in biological terms. One individual does not change its genetic traits within its lifetime. Populations evolve. So a group of organisms can change the frequency that a certain trait appears based on changes in reproduction and survival. But organisms themselves do not evolve. One singular organism cannot evolve. We measure evolution as the change of a frequency of a trait within a population. So make sure you write this down. Populations evolve, individuals do not evolve. All right, so again, genetic variation can come through mutation, genetic drift, sexual selection, artificial selection, and I wanted to really quickly introduce you to the peppered moth scenario. So this is a classic example of natural selection and variation and how a changing environment can actually affect the success of particular organisms. So the peppered moth is one species,
And within this story, there are types of peppered moths that have sort of this lighter coloring, a little speckle peppered, as you see, and then there are variations that are darker or black. Now, at first, the ones that were more successful were the ones with this peppered coloration because they could blend in easily and camouflage on the bark of the trees where they liked to sit. And the ones that were this sort of darker color or solid color were easily spotted by predators, and there were not as many of them being successful within the population because they were easily captured and attacked and eaten. Now, when the Industrial Revolution occurred, a lot of the trees became dark with soot from different industrial plants, and guess what happened? The organisms or the versions of the peppered moth that were darker were more likely to survive and reproduce because the lighter ones could then be spotted by predators. So what we saw was a change in the frequency in the population of the organisms that were able to survive and reproduce.
The concept of biological evolution uses lots of different types of evidence in order to support our understanding of it.
Now we can use other biochemical evidence to help us determine which proteins and DNA are similar in different organisms. This helps us construct our tree of life and gathers more evidence for evolution, helps us figure out how different organisms are related to each other. We can also use embryonic development, fossil evidence, or morphology or different physical characteristics to determine which organisms might be related and where they came from. We know that Earth's present-day species developed from earlier distinct species. When we get new combinations of existing genes or mutations in genes, and these are inherited, they can be passed on to offspring, and we can see variations in populations. For example, if there's a random mutation in this DNA where this A is changed into a G, this could then produce a different amino acid, which could then lead to a different function of a protein and give us a different trait. Mutations can be caused by different mutagens like radiation or chemicals, but they can only be passed on to offspring if they occur in sex cells. If we have mutations occurring in body cells, they're only going to be passed to other body cells that arise from the mitosis or the cell growth of current cells in the body. Over time, organisms that are better adapted to their environment are more likely to survive and reproduce and pass on their genes to the next generation. For example, if we have a population of bacteria and some of those bacteria are naturally resistant to antibiotics, when we treat that population with antibiotics, only the ones with the resistance will survive. Then that gives way to give the opportunity to the resistant bacteria to survive and reproduce and pass on the resistant gene to their offspring. And what we've done here is created an antibiotic-resistant population. The resistant bacteria were more fit, and they have survived and reproduced at a greater rate than non-resistant bacteria. When there is more variation within a species, and a species is more genetically diverse, they are more likely to survive when there's changes in the environment. When we have different types of adaptations, these can lead to the survival of different organisms. We know that behavioral adaptations, structural adaptations, and reproductive adaptations have helped different organisms survive through hundreds and thousands and millions of years. Remember that there's no one ultimately evolved organism. Environments are constantly changing, and so populations will continue to change. Also remember that individuals do not evolve, populations evolve, and evolution is just the change in different frequencies of different alleles over time. Sometimes we can have extinction of a species when we don't have adaptive characteristics that are sufficient for survival. We do see a lot of extinction in the history of life on Earth, and most of the species that have ever existed on Earth are actually now extinct, and we can tell this by the fossil record.
Now, what is the Hardy-Weinberg equilibrium anyway? Well, Hardy-Weinberg is actually an equation that we can use to determine the frequency of alleles in a population at a given time. Hardy-Weinberg is used as a model for describing and predicting allelic frequencies in non-evolving populations. Remember, it represents ideal populations that aren't changing. Since natural populations do change, Hardy-Weinberg provides us with a baseline to gauge that degree of change. Now, there's two equations you're going to need to know in order to do Hardy-Weinberg problems. The first one is easy. It's just P plus Q equals 1. The second one is P squared plus 2PQ plus Q squared equals 1. Now, what do each of these things represent? So P is going to equal the frequency of the dominant allele in a population. Sometimes we represent that with a capital letter, like big A. Next up, Q is going to represent the frequency of the recessive allele in a population. So for example, little a. P squared is the frequency of homozygous dominant genotypes in the population. So for example, big A, big A. 2PQ is going to be the frequency of the heterozygous genotype in the population. So big A, little a. And then you guessed it, Q squared is going to represent the frequency of the homozygous recessive genotype in a population. So if you're confused already, just make sure you come back to this key so you know which letters are going to represent which genotypes and allelic frequencies. The best way to do Hardy-Weinberg is to actually practice it and see it in a practice problem, so let's go ahead and get started. This practice problem comes first. In a population of humans, 16%, or a frequency of 0.16, are not able to roll their tongue. We're going to deal with frequencies to do these calculations, so you want to convert percentages to frequencies, or that decimal version of the percentage. In the population, 16% are not able to roll their tongue, which means they are homozygous recessive for tongue rolling. Using the Hardy-Weinberg equations, determine the frequency of homozygous dominant tongue rollers in the population. So in order to figure out how to solve this problem, we need to know what it's asking. The frequency of homozygous dominant genotypes in the population, if we look back at our key, is p squared. So we're solving for p squared. We already know something in this problem though. So the one variable we already know is q squared in this situation. That is going to be a frequency of 0.16. The next variable we know we can figure out by just square rooting q squared to get q, and that'll give us 0.4. Now we can use that first equation, p plus q equals 1, to determine p. See, now we're just doing basic algebra to get the variables that we need. So in order to determine p, we rearranged some of the variables to solve for p. And 0.6 squared is 0.36, and that is our value for p squared, or the frequency of homozygous dominant tongue rollers in the population. Let's look at one more easy practice for today. Let's say we have a population of rabbits. Some are brown and some are white. The brown allele is dominant, the white allele is recessive. So brown rabbits can have big B, or big B, little b. White rabbits can only be little b. Know that the frequency of the big B genotype is 0.35. So let's try to figure out the frequency of heterozygous rabbits, the frequency of the big B allele, and the frequency of the little b allele with this information. You know that the frequency of the big B genotype is p squared. If p squared is 0.35, we can take the square root of that and get 0.59. From there, if we know p, we can easily solve for q. After we solve for q, and we're looking for the frequency of the heterozygous rabbits, all we need to do is get 2pq. So 2 times p times q. That's 2 times 0.59 times 0.41 would be about 0.48. The frequency of the big B allele we already found, that's 0.59. The frequency of the little b allele is 0.41. So phylogeny is the study of evolutionary history and how organisms are related to each other. And a lot of what we talk about in evolutionary history and phylogeny is the idea that all organisms come from a single common ancestor. Now, when we talk about using phylogenetic trees, which are models to show these evolutionary relationships, sometimes we're actually going to be using cladograms. You might have been taught that cladograms and phylogenetic trees are the same thing. There's actually a few differences between them, but knowing all the precise differences between the two is not something you're going to encounter in high school biology or even AP Bio. So a cladogram might look like something you see here that's depicting the evolution of different types of plants, or it could be very complicated like this phylogenetic tree we see on the other side, which is all the members of the Mycobacterium genus and how they're related to each other based on biochemical evidence. Usually in a phylogenetic tree, this is the branched one you see right here, the distance between two individuals is going to relate to time and how closely related certain species are. Cladograms, on the other hand, are mostly based on shared traits, not DNA or other biochemical evidence, and they're not going to be drawn to scale. And not all phylogenetic trees are drawn to scale either. But for the purposes of this video, we're just going to be talking about evolutionary trees in general, and you'll be able to see them, how to interpret them, and how to create your own. So like I said, these are tools or models used by scientists so we can compare evolutionary history or relatedness between different organisms. So if it's a simple cladogram like this, we're going to be grouping organisms based on the things they have in common. And usually when we're creating trees, if we're going to be doing this on our own, we want to create one that is the simplest explanation for how these traits could have evolved. That's called maximum parsimony, and it means that in most cases, a trait will usually evolve just one time instead of multiple times on multiple different branches. So if we go by that, then we want to create the simplest possible explanation for how these organisms could be related to each other. And don't worry, you'll see what I mean in just a little bit. So in the past, phylogenetic trees used to be created just on shared traits or morphological features, physical features that organisms had in common. Now we use lots of biochemical DNA evidence, similarities of different proteins, so the amino acid order in proteins of different organisms. And that's the data that's going to go specifically into a phylogenetic tree. And we continue to refine these trees as we gather more evidence. And when we say they show relatedness, we're showing how recently two different species groups had a common ancestor. So if we look at this tree right here, again, ignoring the branch lengths for now, we see that each end point on the tree, A, B, and C, that's going to represent a different species group. And the last time on this tree that A and C shared a common ancestor was at this branch point here on the tree. Sometimes on trees, you'll also see these synapomorphies or shared derived traits. And these can be marked by little hash marks on the tree. And any organism that appears after that trait is marked on the tree has the trait or continues in their evolutionary history with that trait. So for example, on these ones, long wings is a trait that both species B and A have, but species C does not have. And a lot of these times, these trees will be rooted, and what that means is there's going to be this sort of continuing branch or part of the tree, and it's actually the root of the tree, and that is in reference to LUCA, or the last universal common ancestor, which we think all living things share. So if we're looking at a tree like this that has this type of branching pattern, remember that continued root, that is in reference to the last universal common ancestor. And then this circle mark here, that is a speciation event, or an event where these two different groups branched from each other. And as time goes on a phylogenetic tree, it will continue from where the ancestor arose. A lot of times these are not drawn to scale along a period of years or millions of years. Okay, so let's look at this plants one more time. You can see now we have an example where these traits appear on the cladogram. And so we see that mosses, ferns, pine trees, and flowering plants are all on this tree, and they all arose from one common ancestor. Now, the last time mosses and flowering plants shared a common ancestor would have been at this branching point down here. And then vascular tissue, this is a shared derived trait that only ferns, pine trees, and flowering plants share. The evolution of seeds occurred here at this point in the tree, so only pine trees and flowering plants have seeds, and then of course only flowering plants have flowers. Everything that branches from before this mark on the tree does not have that trait. Scientists classify organisms into different groups or categories, and this includes all organisms that are currently living today that we know about, and every organism that we've discovered from the past. And they base these categorizations and hierarchies on similarities in their morphology or physical structures, on their developmental stages, so embryology, and they use information like biochemical data, but we can draw conclusions about organisms that lived in the past based on data we collect from the fossil record. Now, there's been many different classification systems over time. It used to be just two groups, plants and animals, and then it got a little bit more specific. In the past, there's been the kingdom structure. Now we organize all living things into three major domains: bacteria, archaea, and eukaryota, or organisms with a nucleus. So let's talk about these domains a little bit more. Bacteria, but bacteria are super prevalent all across the globe, and most bacterial species don't cause disease. And there's a lot of them that are really beneficial, that help us create food like cheeses and yogurt, and others that help us produce medicines. Archaea is its own domain, and these are also prokaryotic organisms like bacteria. They do not have a nucleus. Many archaea are known for being extremophiles, meaning they live in really extreme environments, or really hot environments that are extremely acidic, or really harsh environments. And then eukarya includes anything with a nucleus, so that's plants, animals, fungi, protists, which we'll talk about in a little bit. So these levels of classification are the different categories that we organize organisms into, and different species may have slightly different levels of classification depending on what you're looking at. Some plants don't go exactly with all of these levels and sublevels, but domain is the largest, and then it goes kingdom, phylum, class, order, family, genus, and species. This is the typical categorization for all living things. And genus and species those are the two most specific categories for the most part. Sometimes we can have a subspecies. And we use these two categories to create the scientific name for an organism. Now, a scientific name follows the binomial nomenclature format, and every organism ever discovered is given a scientific name that is two words. The The first word comes from the genus group, and the second word comes from its very specific species group. So its full species name or full scientific name is those two parts put together, the genus and the species. And so it's always two words, unless we have a subspecies, and it's always kind of Latin sounding. It's always in italics. The first word is capital, and the second word begins with a lowercase letter. So some common organisms you might know, here are their scientific names: Gallus gallus, that's a chicken; Sus scrofa, that's our boar here; Entamoeba histolytica, that is an amoeba; and Toxicodendron radicans, that is poison ivy. And anytime a new organism is discovered, they are given a new scientific name. What's cool about these is that they are the same all across the globe. Everyone can identify, for example, a chicken as Gallus gallus. So we don't have any language barriers there. Now, within eukaryotes, or organisms with a nucleus, animals is probably one you're very familiar with, plants, fungi, and of course, protists. They used to be a kingdom themselves, but they're actually so different that we have a bunch of different categories for them. And so going backwards through these categories, protists are usually unicellular. There's some multicellular protists like kelp, but they are eukaryotic, so they do have a nucleus and other complex organelles. They have a bunch of different adaptations for movement, like cilia or pseudopods, as part of their cellular structures. Fungi can be unicellular, like yeast, or multicellular, like these mushrooms here. And they are organisms with cell walls, so each of their cells has a cell wall like plants do, but unlike plants, fungi have chitin, which is a special material that helps it maintain its structure. All fungi are heterotrophs, meaning they have to get their food from another source. They can't produce it themselves. And so fungi typically are decomposers. They can also be parasites as well, and so they'll break down dead organisms for their nutrition. Plants are autotrophic, so these are typically green. They will be performing photosynthesis in order to get the food that they need, so they use sunlight energy and convert that into glucose or other organic compounds. And within the plant kingdom, we have things like mosses and ferns, gymnosperms or conifers, and angiosperms or flowering plants. And then, of course, animals here, these are multicellular organisms. They do not have a cell wall, they are heterotrophic, and they consume their food in a variety of different ways. They can be herbivores or carnivores or omnivores. So over time, there have been many theories about how life first appeared on this planet, but some of the most important experiments that have been done to figure out what was going on include Louis Pasteur's experiments that disproved spontaneous generation, or the idea that life could just appear out of nowhere. And his actual version of this experiment happened with chicken broth and an S-shaped flask to show that bacteria couldn't just appear out of nowhere either. In 1955, one of the most important experiments in our understanding of how life could have arisen on this planet was done by Miller and Urey, and they predicted that if energy was introduced to certain elements and gases, such as a burst of lightning, so they replicated that with electricity, that organic compounds could appear. And they were successful in generating the precursors to some of our organic compounds that are essential to life itself. Now, they didn't create life in this experiment, but they formed precursors to these molecules that are the basic ingredients for life. So, through this experiment, we were able to support the idea that organic compounds could have formed in the conditions that were present on early Earth. So LUCA, like we mentioned in the last video, the last universal common ancestor, is this hypothetical organism that's believed to be the common ancestor of all life on Earth that arose billions of years ago. There's lots of evidence that unifies all living things. For example, all living things have the universal genetic code containing DNA or RNA. We see common metabolic pathways like glycolysis in virtually all living things, as well as some physical similarities in the shapes of cells across all domains. Remember that we have prokaryotic cells without organelles or membrane-bound organelles and eukaryotic cells with them. And scientists believe that eukaryotic cells arose through a process called endosymbiosis. So this theory of endosymbiosis describes how larger prokaryotes could have engulfed smaller ones, and those smaller prokaryotes formed a symbiotic relationship with the larger ones and later became things that we know as the chloroplasts or the mitochondria. And this is supported by the fact that the mitochondria has its own DNA and its own membrane and could have once been a prokaryote itself. Okay, let's go through a brief timeline of some major events in Earth's history. I'm not going to hit on the 5 major mass extinctions or some of the major geologic areas in this video, but that is definitely something worth reviewing if it's covered in your biology class. I just want to mention also too that the timeline of life on Earth is huge, and this is not representative of all the major events that occurred in the evolution of different species on this planet. These are just some interesting things that happened at different points along Earth's timeline. So about 4.6 billion years ago, Earth was was formed, bombarded by meteorites and comets. And then around 3.8 billion years ago, we think the first replicating molecules, which were the precursors to RNA or DNA, first formed. Then we think around 3.5 billion years ago, our first unicellular life evolves, and we have, for the first time, photosynthetic bacteria releasing oxygen into the atmosphere. But it's not until around 555 million years ago that we have many multicellular organisms in marine environments and diverse multicellular life in the oceans. Around 450 million years ago, we see arthropods moving onto lands that would later become things like our land insects, mites, millipedes, spiders, scorpions. 420 million years ago, land plants first evolved. So it's interesting to see that that is later than our first land arthropods. And then 225 million years ago, we have dinosaurs and mammals on Earth, and 65 million years ago is when the KT extinction occurs, wiping out the dinosaurs on Earth. So they go extinct and birds and mammals are able to survive. And then 130,000 years ago, we see the evidence of the first anatomically modern humans on planet Earth. Now we know that all organisms need energy to survive, to maintain their structures, to grow, to reproduce. But different organisms have different ways of obtaining or harnessing that energy. So a lot of what we talk about when we talk about energy in ecosystems is how the energy can flow through ecosystems in different ways. Now we'll also be talking about how matter can flow through ecosystems, and that's what food chains and food webs can show us, is the flow of matter and energy between different organisms in an ecosystem. And we're going to begin to see how all organisms are interconnected and how, for example, a change in the number of producers in a particular ecosystem can affect the consumers at the secondary, tertiary, even quaternary consumer level. Let's take a closer look. Remember, life is organized. It's one of our characteristics of life. We know that a group of organisms of one species in one location is a population, and populations can be categorized by functions that they serve. Right here, we're looking at a food web. Organisms are characterized in different trophic levels or energy levels depending on where they feed and what they consume. Producers can generate their own food, usually from sunlight energy, sometimes chemical energy as well. Consumers consume the producers, and decomposers break down organic material from other organisms. A food chain shows one pathway of energy flow. A food web, like you see here, can show the interconnectedness of many food chains within a particular ecosystem. Keep in mind that on a food web, the arrows always point towards the consumer or the organism that is doing the eating. So here we have the rabbit arrow pointing towards the cougar or mountain lion, which means the rabbit is getting eaten by the cougar or mountain lion. He is doing the eating, so the arrow points into his belly. Important word here is an autotroph, also known as a producer, but these are organisms that can make their own food or are able to form their own organic molecules for food. Plants are autotrophic. Heterotrophs are organisms that must consume other organic compounds for food, and that means consuming other organisms. Heterotrophs are consumers. For example, humans are able to obtain nutrients by consuming other organisms or the products of other organisms. This is a typical trophic or energy pyramid that you might see. We have our producers or our autotrophs at the bottom. These are organisms that are going to get energy from the sun through chemical processes, photosynthesis, and then on the next level up, those are the primary consumers, the organisms that consume them. The next level up, we have the secondary consumers, tertiary consumers, and quaternary consumers. There's different types of pyramids too, like pyramids of biomass and pyramids of numbers. But here, what we're looking at are these different trophic levels, or levels that organisms feed at. And we want to keep in mind that at every level, we have a significant amount of energy lost. So it is inefficient. We have about a 90% energy lost every single time we go up a trophic level. So it's more efficient energy-wise to consume at lower trophic levels, but only about 10% of that energy is retained every time you go up a trophic level. I want to go back to talk about how all energy on Earth is really supported by the sun. And of course, all matter here on Earth is here to stay, and the atoms and molecules that we interact with and make up our bodies are the same atoms and molecules that were here hundreds of thousands of years ago. Think about how carbon, hydrogen, nitrogen, and oxygen all combine and recombine together and are passed through different food and matter systems. But analyzing the flow of energy and the cycling of matter within an ecosystem is an important part of biology. So this diagram is something that you might want to consider. These plants are going to use carbon from the atmosphere to undergo photosynthesis and create glucose and oxygen, which are our ingredients for respiration. But if we think about if more snails are added to this container, what effect is it going to have on the plants in the container? Eventually, if we add more snails, we would see more carbon dioxide, dioxide, and more carbon dioxide would lead to more plant growth because the plants could take in more carbon dioxide to perform more photosynthesis. Now, there are lots of ways that matter and energy are cycled throughout the planet. The carbon cycle is one example of that, and there's lots of different processes where carbon, that element, is converted or passed between different organisms or between organisms and the environment. So from the air, plants and other things like phytoplankton in the water can take in carbon as carbon dioxide and use that in the process of converting sunlight energy into organic compounds. So the carbon dioxide in the air goes directly into those organic compounds or things like glucose. And then all living things perform respiration, but in the process of respiration, we use that glucose to generate ATP, which is another molecule organisms use for energy. So that process is respiration. So if we follow the path of carbon here, we say that carbon can come from the atmosphere into photosynthetic organisms like trees and then can be consumed by other organisms. So when animals eat plants, they're taking in carbon that way, and then they're exhaling carbon dioxide after the process of respiration. Now, not all the carbon leaves their bodies. Eventually, they will die, and after death, the carbon can be returned to the environment through the help of decomposers who will break down the carbon in dead organisms' bodies, and that decomposition process will release carbon either back into the soil or back into the atmosphere. Another way carbon can enter the atmosphere is through combustion. So the combustion of fossil fuels also releases lots of carbon into the atmosphere. And when there's lots of carbon in the atmosphere, it actually can be taken up by the waters in the ocean. And sometimes we have lots of carbon going into oceans, and that carbon actually turns into carbonic acid in solution in water, which leads to lots of ocean acidification. So the more carbon in the atmosphere, the more the oceans can take in, and the more acidic the oceans get, which is not always great for the ecosystems there. Now, if we go back to our levels of organization, just as a reminder, remember that a population is a group of organisms of the same species living in the same area. Typically, community is one level up or one level of organization greater than population, and it involves all the species that interact and live together in a particular ecosystem or habitat. When we get to ecosystem, that's when we're talking about the biotic and abiotic factors together, but think of population as one species, community, several different species interacting together, and then ecosystem, all of those species plus the non-living factors as well. So today we're going to be talking about factors that influence population growth, different ecological relationships. You might have heard of types of symbiosis like mutualism, parasitism, commensalism, and of course the role of different non-living or abiotic factors. In one ecosystem, we have lots of biotic, living, or abiotic factors. And an ecosystem is shaped by the interactions between these non-living and living factors. In every single environment, there is competition between different organisms and interactions between different populations. And often within one ecosystem or environment, there are natural checks in place that keep populations and ecosystems relatively stable over hundreds or thousands of years. Though sometimes we might see certain individuals that have what looks like unlimited or exponential growth, most of the time, most populations will reach what we call as a carrying capacity within a particular environment. Because there are limited resources within a particular environment, like food availability, water, space, at some point those organisms are going to reach the maximum population that that ecosystem or environment is able to sustain. And even though that may fluctuate over time, a population may be relatively stable once it hits its carrying capacity. If we see a sharp decline like this, it's probably because there was an introduction of a disease, or a new predator, or an invasive species. All of these biological molecules can be limiting as well. So the number of organisms any particular habitat or ecosystem can support is dependent on the availability of certain resources within that environment. For example, energy, water, oxygen, nutrients, all of those are going to have an effect on how many organisms an environment can support. In general, there's lots of factors that are going to influence the growth of the population, but we'll see growth typically when the birth rate is larger than the death rate of a population. If we don't have any limiting factors, a population can grow exponentially and it'll grow in that kind of J curve. But if we have some sort of limiting factor or a particular resource that's going to limit the population growth, we'll see a logistic growth curve. So it'll increase until it reaches that carrying capacity and kind of level out. There's also a lot of other factors like community structure, interactions, energy availability that are going to determine how those populations will continue to grow and survive. This question just gives us a random population and shows us how the population is growing over time. In this graph, you'd have to estimate where the population's growth rate is the highest. Well, it's going to be at a certain point in time. Can you see where the slope is the greatest for this particular population? That's where the growth rate will also be the highest. Okay, when we talk about the interactions of communities though, there's different types of interactions like predator-prey or predation interactions, but we also have competition, different types of symbiosis.
Organisms can interact with each other in various ways. Mutualism, for example, is a type of behavior where both organisms are going to benefit. So you might think of things like a bee and a flower. Both are getting advantages here. The bee is being able to get the pollen in order to create the honey it needs. The flower is helped with its reproduction by the distribution of pollen. Another type of symbiotic relationship is parasitism. This is where one organism benefits and the other is hurt or harmed. Mosquitoes, and of course the protist malaria is a parasite as well. Commensalism is a type of relationship where we have one organism benefiting and the other is neither hurt nor harmed, and these are things like barnacles that attach to a whale. Now, there's also predator-prey relationships, which can be very dynamic, and predator-prey populations can fluctuate depending on what's happening within the ecosystem. In terms of communication and behavior, a few terms you might want to know. Imprinting is when a young animal learns something specific in a particular period of time. Territoriality is where there is a protection of a habitat or territory with tactics such as aggressive behavior, songs, pheromones. Conditioning is a process that leads to learning, and we have both classical and operant conditioning. And habituation is going to be when an organism is going to show a decreased response to a stimulus over time. And ecosystems can be relatively stable over hundreds or thousands of years, even though populations within the ecosystem may fluctuate depending on certain factors.
So biodiversity in general is just the variety of life on Earth. There are countless species literally on planet Earth, and so many we have not discovered. But a lot of times when we talk about biodiversity, we're talking about species biodiversity, which is a measure of the variety of different species in an ecosystem. So an ecosystem with very few different number of species has low biodiversity, and an ecosystem with very many different species has high biodiversity. We can also talk about things like genetic biodiversity, which is the genetic differences among individuals within the same population. But for the purposes of this video, we're going to be mostly talking about species biodiversity when I say biodiversity. The higher an ecosystem's biodiversity, the more resilient it is to changes in the environment. And so we often say the more biodiversity an ecosystem has, the healthier it is. Ecosystems with lower biodiversity and just fewer parts to them are often more susceptible to things like disease. When one disease wipes out a particular population, if there's only a few species in that ecosystem, it could have devastating effects on the entire area. These areas are often less resilient to other changes as well. So why do we care about biodiversity besides saying that an ecosystem with higher biodiversity is healthier? Well, mostly it's due to the fact that humans rely on other organisms for almost everything, for products, for medication, for the supplies we need to build and construct things, timber, fibers, adhesives, rubbers. All of these products come from things that were once living. Many prescription drugs that have been developed come from wild species, and there's so many more species that haven't been studied that have huge medical potential. Other species could be indicators of changes within an environment, of toxins, for example, and when we notice if population levels of certain species rise or fall, this could be an indicator of bigger problems within an ecosystem. Now we know that every species occupies a particular niche within an ecosystem or specific role that it plays, but some organisms may have a greater impact on a particular ecosystem and its stability than others, and so we call these often keystone species. The wolves from Yellowstone Park are often used as an example of a keystone species, even though there's been some debate recently about other organisms that may be just as important to the balance of that particular ecosystem. In general, we know that climates with warmer and wetter conditions have higher biodiversity, so abiotic factors can influence the biodiversity of an area as well. So if you think of a typical rainforest with very high temperatures on average throughout the year and high levels of rainfall, these typically have the highest levels of biodiversity on the planet. Then you compare that to something like the Arctic tundra. While not completely barren, there's plenty of species that are able to survive in the tundra. It's fewer than the rainforest, which has conditions that support a greater variety of life. Now, there are lots of different ways to measure biodiversity. There are different biodiversity indices, or a biodiversity index is one way to quantify quantify the levels of biodiversity in area. And of course, we can't go and count every single species in every area. So often what scientists do is ecological sampling, where they use a portion or a piece that's a representation of the whole ecosystem. But let's just take a look at this real quick. These are hypothetical species represented by dots. Every color represents a different species. And in each of these ecosystems, we have 24 different individuals. So to calculate biodiversity for each of these, we would take the number of different species and divide it by the number of individual organisms in the ecosystem. So here we just have one color, so that's one specific species out of 24 different organisms. In the second environment, we have 7 different colors out of 24 different organisms. And here we have 9 different colors out of 24 different organisms. And here we have 5 different colors, which means 5 different species out of 24 different organisms. So if we did the math, we would see that this ecosystem here had the highest biodiversity using this particular calculation. Now, if you continue to study biology, you look at it in college, or get into AP Environmental Science, you'll see that there are other ways to calculate biodiversity. So Simpson's biodiversity index is one, Shannon-Weiner is another. There's even calculations for species evenness and species richness in an area, and all of these serve a different purpose for different scientists looking for different things. So let's get back to these hypothetical different ecosystems here, and let's just see an impact of a disease on one species in particular. So let's say a species is impacted by a disease. It starts with one individual here, and so that line crossed, that's going to represent the individual that's affected by the disease. And what's going to happen is that disease might spread to any individuals that are within close proximity to the original infected individual. And so what happens is that individual dies, as does any of them of the same species in that same. And so any of them that are touching or surrounding this individual that first got infected will die off as well. So let's see what happens to those ecosystems. So our first ecosystem entirely wiped out. There's just one species, all of the individuals are dead. This particular ecosystem is pretty heavily affected as well. These two did not come out so bad. So this one here, although it did have a number of individuals of that green species, we can see that it was also surrounded by many different types of species. So the disease did not get as far, and we have a lot of surviving individuals, as did this ecosystem here. So these dots are just a little hypothetical, but when we look at real life ecosystems, the dots could be stand-ins for tree species. And so we've seen situations similar to this happen in real ecosystems where we have trees, like conifer, for example, it's affected by a particular disease, and then the conifers in the ecosystem are all wiped out because the disease can travel from individual to individual very quickly. So when we talk about these changes that ecosystems are resistant to, some of them can be natural, so they can be droughts, flooding, earthquakes, other catastrophic events, wildfires, climate changes, in addition of excess nutrients to a system, which is eutrophication. All of these are large-scale changes that can influence the biodiversity of an ecosystem. And in tomorrow's video, we're going to be talking more specifically about the human impacts and disruptions to ecosystems that are caused by humans. Earth's organisms share limited resources. We rely on processes like the water cycle, the nitrogen cycle, the carbon cycle to cycle out essential nutrients and molecules in and around the Earth that are essential to survival. And of course, the Earth changes over time, but humans are a huge source of environmental change. And if we look at this graph here on screen, I'll blow it up for you so it's a little bit bigger. This is the world population growth in billions. It's a little outdated because we know now that the world's population is estimated to be above 8 billion, but the depletion of Earth's resources and different impacts from humans and their behavior is often a direct result of human population growth. And often humans are the cause of major disruptions to different natural ecosystems. And because we rely on the natural world and organisms and things found in nature for products and for medicine and for our own survival, making sure that we protect Earth's resources and its organisms is important to our own survival. Now let's talk about these resources that are found on Earth. So renewable resources are any natural resource that can be replaced by natural processes relatively quickly. When I say relatively, generally that means within a human lifespan or at a rate faster than the rate that they are used. Non-renewable, on the other hand, cannot be replaced within human lifetime. Now, non-renewable resources come from natural processes like fossil fuels, for example, but it takes millions and millions of years for these resources to form. So when we're speaking in realistic terms of humans and what we have access to, once non-renewable resources are used up, they are gone. There's not going to be any more for a long time, and they may not even replenish. They are a limited resource and they may be used up by humans. So examples of these are coal, oil, natural gas. Renewable, on the other hand, we could talk about things like wood, organisms that we use as resources like fish. But it's important to note that renewable resources can become non-renewable if humans use them up too quickly. Now I want to talk a little bit about the ozone layer. Now this is an interesting case of human impact, and the hole in the ozone layer we know is primarily caused by CFCs or chlorofluorocarbons, which are chemicals that come from things like old aerosol sprays, and these have actually made an increasing hole in the ozone layer. Now the good news is that after mitigation, after some laws that went into effect banning CFCs in certain products, we have started to see a decrease in the hole in the ozone layer, which is really good. But this is the progression of the hole from 1979 to 2008. So you can see in these images, they're getting bigger. But in the recent years, we have seen data to show that this hole is shrinking. So that's some good news. But the ozone layer protects Earth from harmful radiation, UV radiation, and we know that compounds in certain products have damaged this ozone layer. One thing students often confuse is that the hole in the ozone layer is a major contributor to global warming. And those are actually two separate issues, so let's talk about both of them. Global warming is an increase in the average temperature of the biosphere on Earth. It's mainly a result of burning fossil fuels and introducing greenhouse gases into the atmosphere, which trap heat at a higher rate than normal. And alongside global warming, we see other issues like rising sea levels, coastal flooding, melting of polar ice caps, melting of glaciers, all of which can have devastating effects on the environments and the people who live in those areas. Now, there are other activities that humans can do that cause disruptions, not on such a massive global scale, but on a scale that can be a huge problem for local ecosystems. So for an example, humans often have either intentionally for a good purpose or unintentionally introduced an invasive species to an area that does harm to the native species in an environment. Now, an invasive species is a non-native species, so something that wasn't originally from that place, that comes and does harm to the local ecosystem. A really common example of this is kudzu in the southeast. It was introduced as a decorative vine, something used in erosion control, and it has completely overtaken local ecosystems, totally covering native plants, blocking out the sunlight, outcompeting native species and reproducing at a faster rate, which then decreases the overall biodiversity of the ecosystem, which we talked about in yesterday's video. Human activities can also introduce new diseases to an area. Elm disease was a tree disease that was introduced, totally devastating, and often we see more of this with the increased movement of humans and human activity on the planet. And habitat change, of course, is a huge threat to different species. Actually, habitat loss is the #1 reason species are at risk of becoming extinct. Habitat loss can occur for a number of different reasons from human activity. So pollution in an area could destroy habitat. For example, certain chemicals or gases could kill off native species that different organisms rely on to survive. This can obviously come from the burning of fossil fuels, but also chemicals, runoff. Even certain fertilizers could end up be damaging to different species in an area if they're carried off into local water supplies. Deforestation, just removing a large amount of trees for a certain area, either for the use in lumber or for building roads or other things. And again, related to that is logging, monocropping, so when you're planting only one specific species in an area. And industrialization is in just any area is a good example of this, but as more nations become industrialized, we see more and more of this habitat change in local ecosystems. So a lot of this sounds like bad news, and when a lot of students hear all of these things that humans do that can disrupt local ecosystems, a lot of times they think, okay, well, I can't do anything about that, or, well, just humans are all bad and everything we do is going to harm the environment. But that's not true. There are other ways, or plenty of ways, where humans can have a positive impact on the environment. So the classic reduce, reuse, so if we reduce our consumption of certain resources, or we recycle and compost our resources, so instead of generating more waste, which causes more problems in environments, if we're composting, we're reducing the buildup of things like methane gas in landfills, and it's going back into the ground where decomposers can do their work and return those molecules into biogeochemical cycles that are healthy for environments. Planting native species is another way that's hugely beneficial for the biodiversity of an area. Instead of planting non-native or invasive species, which could end up causing harm to a local ecosystem. Of course, supporting sustainable practices. A lot of this is out of our hands as individuals, but we can support companies that have sustainable practices or vote for legislation that we know will support the environment. And of course, advocating for environmental policies. Like I said before, when we banned the use of CFCs in certain products, that actually did have an effect. We stopped increasing the hole in the ozone layer, and it is ending up starting to heal. If you're interested in these topics, I would recommend looking into an environmental science, like AP Environmental Science course, or if your college or university has has topics on this. Environmental studies are a really fascinating and important key area of science that will affect our lives and the lives of those in the future. That is it. Thanks so much for watching. You've gotten through an entire year's worth of biology content in just a little over 2 hours. Good luck with your studying and your review. Thanks so much for watching. Give this video a like if it's been helpful, and I'll see you later.